All right, let's go ahead and get things started for the day. Yeah, thank you, Stu. Quick reminder to get on Discord, and we've got a chat channel that has links to voting. So vote for the best short talks as well as the best long talks. And we will have some awards at the end of the day. Okay, so with that, I would like to, it's my pleasure to introduce my friend and colleague, Dr. Vicki Webster-Wood. We worked on our PhDs together at Case Western Reserve University. So she was there and got 3 degrees, her BS, MS, and PhD. And she also transitioned, so we did that all under the direction of Dr. Roger Quinn. And from there, she transitioned to a postdoc where she focused on tissue fabrication and mechanobiology. And after that, she moved to Carnegie Mellon University, where she is now assistant professor in the Department of Mechanical Engineering. And she leads the CMU Borg Lab. And she's been one of my favorite people to ever collaborate with. And I'm always interested to hear her ideas and perspectives on different topics. And today we're going to learn from her about beyond biohybrid systems.
Thank you, Alex. So I'll go ahead and get my screen shared and make sure we're seeing the right screen. Yeah, not seeing slide notes.
Yes, all okay.
Okay, excellent. So thank you again, Alex. It is a tremendous honor and a special privilege to be joining you all here today for the final plenary session of Living Machines 2022. This conference holds a truly special place in my heart. This was one of the conferences that growing up, if you will, in Roger's lab, we went to almost every year, and I've met a lot of really fantastic academic friends and colleagues through this community. And while we can't be together this year in person, I'm honored that you all have joined this virtual space at whatever time of day it is where you are to join in this conversation. I think this community shares a really important vision and mission for robotics in which we seek to understand biology and create new bio-inspired, biomimetic, and biohybrid systems. I thought a lot about how I wanted to approach this talk this morning. We've seen some really fantastic plenaries this week from Dr. Thiel and Dr. Kovac, and honestly, it's a little intimidating following that up as an assistant professor. And while I don't have the breadth and depth of research experience yet in my own group, which is relatively young here at CMU, I really do want to share kind of a future-looking vision for the long-term development of bio-inspired robotics. So over the course of my about 45 minutes, my goal is really to convince all of you in the audience of the need to think about moving beyond just bio-inspired and to consider how we can build renewable and sustainable robots by using biomaterials and other living materials in these systems, kind of building on the theme of sustainable robotics that we heard from Dr. Kovac yesterday. And make sure my clicker is clicking. There we go. Okay. For many of us, when we think about building our robotic systems, we look to nature for inspiration. And in doing so, we see that animals are capable of tremendous feats of agility, ingenuity, creativity, and learning. They adapt and respond to dynamic changes in their environment. They learn from experience, and they solve complex real-world problems. And as robotics researchers, in particular in bio-inspired robotics, these are abilities that we want to capture in our future robotic systems. systems. Over recent decades, robotics has really seen kind of an explosion in both the physical systems and artificial intelligence and controls. Indeed, robots are becoming almost ubiquitous in our built spaces. Of course, this is perhaps most common in manufacturing, where many manufacturing industries have even transitioned to almost fully automated plants. But robots are also entering our homes, right? Many of us may have robot vacuums or smart cars, and drones are now commonly commercially available, right? So we're seeing robots more and more and more in our everyday spaces. As researchers, this means that we are also developing novel state-of-the-art robotic solutions for a broad range of target applications, from the depths of the ocean, where aquatic robots are being developed to perform monitoring and maintenance tasks, to agriculture, where we're developing robots capable of harvesting fruits and vegetables, to even our personal lives in the form of prosthetics, and beyond into space exploration. These developments come with tremendous robotic adoption in the real world, which has seen phenomenal growth even in the last decade, right? So I wanted to take a step back as robots enter our built and natural spaces at increasing rates, and take a moment with this community to think about the materials that we are using in these robots. At a high level, we can break robots down into some kind of key components, right? Most of our robots are requiring some sort of chassis. Of course, we need actuators to move the robot and to kind of generate actual locomotion. We need to be able to sense the environment that the robot is in and control the robot through electronic systems. In digging into the materials we use for these components, we see several themes turning up over and over again in traditional bio-inspired robots, right? There are many metals and plastics in use. Actuators may require magnets or elastomers or ceramics. And in terms of sensors and electronics, we of course have materials like copper, lithium, platinum, and unfortunately, surveys of, in particular, consumer electronics even show levels of hazardous compounds like cadmium, lead, and chromium at often higher than regulator accepted levels. All this is to say that many of the materials we use in robots are non-renewable. Unfortunately, many of the material resources required in modern consumer electronics also face production peaks within the current century, because there's limited availability in natural reservoirs. Right? Some projections put the projected production peak for lithium as early as 2037. This is happening in parallel with a renewed demand on these materials, which are often difficult to recycle. Unfortunately, as demand increases and we keep extracting these materials from our natural ecosystem, the grade of ores that is available also decreases. So the quality of these materials is decreasing, even as their availability is going to decrease. To obtain these materials, not only does mining produce hazardous runoff that can affect nearby groundwater and watersheds, but many mining sites that support the global electronics industry exist in areas with fewer worker protection laws. You may remember in 2019, when many tech giants were sued due to the tragic deaths of children workers in Congolese cobalt mines. Accidents, injuries, and fatalities are not uncommon in support of these non-renewable materials. And some mineral reserves are concentrated in relatively few regions. For example, 70% of the world's lithium is localized in the 10 largest deposits that we've found to date, with an overwhelming share located in just Chile and China. And 98% of the current rare earth element supply is located in China. This means that local disasters and tragedies can have tremendous effect on global supply, which we've seen most recently in the supply chain disruptions caused by the COVID-19 pandemic. The reality is that these natural disasters and supply chain interruptions are only likely to increase over the coming century. So that's what goes into our robots. If, as responsible researchers, we take a moment to consider the cradle-to-grave life cycle of these devices, we must also think about the impact of our robots during deployment and beyond. Well, what happens after we deploy them? Many of us know through painful personal experience that no plan survives first contact with the enemy. And in this case, the enemy is the flat ground. It is not uncommon for robots to be damaged during deployment. And I even recently learned that in some deep sea aquatic robots, they are sometimes even deliberately designed to leak small amounts of oil at tremendous depths in order to protect the electronics. And that's oil entering our ocean ecosystems. Small amounts, admittedly, but I want us to be aware of this and thinking about this. After deployment, our robots, especially in research, they may be part of a student project, and the student may graduate. The luckiest of such research robots may be recycled for its parts, but many may end up ultimately contributing to e-waste. And if we've done our jobs as robotics researchers correctly, our robots should become obsolete, right? We always want to be advancing the state of the art. Perhaps some of these will end up in robot museums at our institutions, but many more may be parted out over and over again until even their individual components are obsolete. Now, I know this is a bit doom and gloom to start the, at least on the East Coast of the US, morning. And I fully acknowledge that consumer electronics, right, our phones, our laptops, our cameras, all of these have a far greater impact on the electronic supply chain and the growing mountains of e-waste than our robotic systems. But since our jobs are so often to think forward and consider the future of the field, I want to pose the following question. What can we, as robotics researchers, do to create renewable and sustainable engineering futures? Well, we could all leave robotics and do research in climate change and renewable energy, but I happen to think robotics is a rather valuable tool, so I certainly wouldn't encourage that approach. Instead, in thinking about this question, I want to encourage everyone here today to look forward, not just to what robots could be tomorrow or in 5 years, but what could robots be in 20 years or 50 years or even 100 years? Many of us in this community are basic researchers and engineers. And we've got many trainees and early career researchers as part of this community. Right? We can think long term. So what can you do now to lay the foundational research needed for robots of the next century to be more sustainable? For me, in thinking about this problem, based on my training in bio-inspired robotics, I look at animals. And I see that unlike traditional robots, the biological systems that inhabit our natural world are already composed of dynamic responsive materials that are naturally biodegradable. For example, tissue growth is guided by these biodegradable protein structures. The muscles that allow us to move are adaptable and self-healing. And you can even think of biological brains as a dynamic responsive material composed of this intricate network of neurons and supporting cells. And this material is capable of complex computation and learning. And recent advances in tissue engineering and microrobotics have started to allow researchers to begin to harness these materials directly into biohybrid robotic platforms. And for the purposes of this talk, I'm really focusing on biohybrid robotics as this idea of combining engineering solutions with living dynamic materials or with biomaterials. For the remainder of my time today, I really want to focus on 3 areas. First, I want to provide a brief historical overview and introduction to the field of biohybrid robotics. Perhaps this will inspire some of you to join the field and we can get our own cluster in Jose's analysis next year. Next, I want to focus in on some of the types of renewable materials currently available in robotics, and in particular on a case study of how basic and applied research with renewable materials, with or without living materials, can be used to bring them closer to deployment in broader robotic applications, right? You can use sustainable biomaterials in robotic systems without having to convert to a tissue engineering lab and grow your own muscle and neurons. So I wanted to focus on that as an example. And finally, I'll conclude with a brief look at some of the challenges and future perspectives on the outlook for renewable and biohybrid robotics as the field progresses. So let's start with a look at this crazy biohybrid thing. Right? In biohybrid robotics research, our research often starts by studying biological systems. We want to be able to learn fundamental principles of actuation and control by studying and modeling biological organisms, and ultimately, even harness the material capabilities of these living systems in future robotic solutions. We need to identify and characterize renewable biocompatible materials and appropriate fabrication techniques. And then if we're going kind of full biohybrid, we can use these materials to build adaptable living actuators and energy efficient living controllers, with the ultimate goal, at least from my own perspective, of creating soft, bio-inspired, renewable, and biodegradable robots that can themselves safely interact with us and our environments. Biohybrid robotics is a young but growing discipline. Even the last decade has seen tremendous growth in the number of publications related to biohybrid robots, biohybrid materials, and biohybrid design. It's an exciting time to be involved in this field. At a high level, we can think of the different types of biohybrid robots. And before I dive into this, I really want to acknowledge the fantastic workshop speakers from last year's iteration of Living Machines in the Biohybrid Robotics Workshop. We've spent this past year coming together and collaborating to think about what is the state of the art in our field and what can be the future of biohybrid robotics. And together we've a manuscript now under review that many of these figures and ideas come from. So at a high level, we can separate the field broadly into kind of three categories. Starting at the smallest scale with single cell organisms, we find microorganism biohybrid robots. These devices are typically fabricated by harnessing the natural motile capabilities of single-celled systems, such as bacteria, right, which can naturally locomote in many aqueous environments. What's really fascinating about bacteria as a medium for robotics is that there's a plethora of genetic tools available to us. And so we can engineer these microorganisms to serve specific robotic functions. We can also use techniques from synthetic biology and cell biology to basically load individual bacterium with cargo, which means that we can use these devices for things like drug delivery, environmental monitoring and quorum sensing. And I've even seen applications where they're using swarms of bacteria for micro manipulation tasks. Of course, bacteria is not the only motile single cell system. And another common system used for microorganism bots is spermatozoa. This is a particularly impactful application for biohybrid robotics because there's direct possibilities to use these robots in the treatment of infertility. In spermatozoa-based robots, there's kind of two approaches. One approach is to use the motile flagella of the spermatozoa to drive your robot. And the other approach is to affix synthetic components to a spermatozoa that would otherwise be immotile in order to allow it to move. And both can kind of be considered in this biohybrid space. Moving up the size scale, we see multicellular systems. Oftentimes, these take advantage of techniques from tissue engineering in order to grow muscle or neural tissue on synthetic robot scaffolds to create locomoting devices. These systems have been used as models for biology For example, like this fish robot, which is driven by cardiomyocytes and provides a model system for studying mechanisms of biological pumping, or can be used in engineering solutions where we can use the tissues themselves as an engineered pump. Of course, this isn't restricted to cardiomyocytes, and a number of research initiatives have used other types of muscle cells, such as skeletal muscle, where tissue can be combined with engineered metamaterials in order to drive alignment and maturation of the muscle cell. Or muscle can even be isolated from insect sources, which provide a more robust tissue for things like operation in air over short periods. All of these tissue engineered based approaches kind of require us to re-engineer the tissues, right? We start from single cell levels, we design scaffolds, and we try to induce the cells to behave the way we want. But of course, nature has already solved this problem. And so we can also consider biohybrid robots based on living organisms. And this is what the field of cyborgs looks to do. In these systems, researchers identify small-scale organisms that already have the locomotion capabilities they want in their robotic platform and combine these with small electronic payloads. This has been done for a variety of applications. For example, for aquatic monitoring, one of my collaborators on this work, Nicole Xu, has shown that you can actually interface jellyfish with these microelectronic electrode nets that allow you to control the swimming of the jellyfish and direct its motion in ocean environments. Perhaps even more simply, you can use the natural abilities of the organism to provide meaningful output. Right? This is an example of a clam that's being used for water pollution monitoring. Clams open and close in response to things they sense in their environment. And so just by kind of affixing this strange little hat to the clam, they're able to get a sensory readout of the opening and use that to infer information about water quality. Cyborg systems have also been shown on larger organisms, such as in controlling jumping or using the natural sensing capabilities of locusts. And of course, many of us are familiar with the fantastic work being done on cyborg beetles, both for walking control and flying control. And similar approaches have been taken on cockroaches. This can be scaled further, including, I think this is the largest example I found so far of a cyborg turtle, in which they used a heads-up LED display to direct the turtle to move certain directions. So there's a lot of possibilities to use a living organism as a platform for our robotic devices. Okay, so these are all examples of biohybrid systems, right, that have living materials. But I wanted to break this down into some of the materials that we actually use in these systems so that you all could try and get an idea of how you might be able to use some of these materials in your robots. To achieve the goal of sustainable, renewable robots, we really need robotic materials that are biocompatible, renewable, and biodegradable, so that we can kind of have that full life cycle of sustainable robotics. I think this is particularly important for applications in biointerfacing, where we want these robots to interact with living organisms or living tissues, and for environmental deployment in sensitive ecosystems. Right? We saw great examples yesterday of the need for biodegradable sensors for monitoring forest environments in Dr. Kovach's plenary. And I think the same can be true for other sensitive ecosystems. One example that commonly motivates my own work is the need for monitoring and maintenance in environments like coral reefs, which are tremendously sensitive. And we don't want to release swarms of robots into these environments that will ultimately be leaching out heavy metals or having plastic bits fall off. Sea turtles' favorite food is jellyfish. And so unfortunately, sea turtles commonly mistake plastics in their environment for jellyfish. And this can be quite harmful for the local life in these systems. And so in biohybrid robotics, we see material solutions for many of the key components that we need in robotic systems. Probably the most common approach in biohybrid robotics is to consider actuators, right? Muscle as an actuator, well, it doesn't exceed any kind of individual metric in engineering actuators, is kind of good enough across the board, right? It meets many of our needs for actuators. And so we see many biohybrid robotic devices that use this material, right? For example, in this system, we see an antagonistic pair of skeletal muscle actuators that are able to drive a robot arm. This can also be used for swimming and other modes of locomotion. As we've seen from the types of biohybrid robotics in the field, of course, muscle is not the only biocompatible actuator or biological actuator, right? Many organisms use flagella or cilia to drive their motion, such as the spermatozoa robots we've discussed. And we can extend this to include other additional structural features for specific modes of locomotion, such as the use of wings and legs, which of course are ultimately muscle-driven without the need for recapitulating the muscle itself. There are also a number of material solutions from biology for the creation of controllers. The kind of fundamental unit of biological control might be considered to be neurons, right? And indeed, we can use cultured neurons to control the locomotion of robots, right? So this is an example of a neural culture. Neural cultures are very sensitive, so they do have to live in special incubators right now. But based on reading and stimulating this network, you can actually drive the motion of small robots, or probably big robots. This one's a small. Right? You can also consider using pieces of small nervous systems, such as brain slices or ganglia. Right? Many of you who were here in 2017 may remember this video of my super slow slug robot, but it's controlled by a small portion of the sea slug ganglia. And of course, with cyborgs, we can use the intact brain of the cyborg organism base in order to perform much of the control and only kind of interfere periodically when we need specific behaviors or motions. So there are a number of approaches to both actuators and controllers that we see in biohybrid robotics. Right? But many of you in the audience, I know, probably have zero interest in joining us in growing our own tissues, and instead need material solutions that you can bring to otherwise traditionally synthetic robots. And I think the biggest area for potential overlap here is in considering the structure. Right? This can be used for things like the chassis, interfaces, anything like that. Right? Unfortunately, this has been kind of a neglected area in biohybrid robotics. So much of our research has focused on actuation and sensing because biological actuators and biological sensors offer so much potential for our robots that's kind of intuitive as you look at these animal systems, right? So I wanted to spend a little bit more time discussing some of the sources of renewable materials that could be used for biodegradable robot structures. We can get these materials from a variety of sources, right? For example, they could come from plants. The emerging field of bioplastics is creating new pipelines for plastic-like material production. And while there's still some research challenges on ultimately being able to recycle these materials, they are available from these kind of renewable or waste sources. And so with continuing research in this space, this could be a great way to get more robust kind of rigid renewable materials for our robots. In the living organisms we so often study, structural materials often come from proteins, right? And we can harness these protein-based materials directly in our robotic solutions. Biological systems exhibit a wide range of material properties that are relevant to robotics. For example, collagen and gelatin provide soft, compliant materials. Silk is flexible yet durable. And keratin and chitin provide a more rigid structural material, like in exoskeletons. So the question then is, how can these biological materials be used in engineering contexts? In order to do this, I kind of see several key system needs. For example, we need to be able to consistently fabricate these materials, right? One of the great things about existing engineering synthetic materials is you buy a piece of steel, you know that piece of steel has the same properties within a tiny tolerance every time you get it, right? We need the same for biomaterials, or at least close enough to be acceptable for engineers. They also need to have well understood mechanical properties, right? We need to have known methods to produce biomaterials with specific mechanical properties so that we can really do forward design. And we need to be able to integrate these materials into useful manufacturing systems. So I wanted to do a little bit of a case study looking at a pipeline that we've taken in my group to transition collagen to a space where we can use it more readily in robotic systems. And hopefully it'll give you a little insight into the steps that could be taken for other materials to bring them closer to this space. For this work, we are using electrocompacted and aligned collagen as a material for biohybrid robots. We didn't develop the electrocompacted and aligned collagen approach. It's a technique that I learned as a student in Dr. Ozan Akas's lab at Case Western Reserve University. As a basic intro, the way this works is that you isolate collagen from skin or tendon, and you introduce a viscous molecular solution of collagen between two electrodes. You apply a current across the electrodes, and the collagen molecules become charged and compact along the isoelectric point. Using this technique, we can fabricate robust collagen threads and sheets. So this gives us a consistent fabrication method for robust materials based on this biomaterial. And the little asterisk here is that it's fairly consistent. I think that many of these biomaterials are going to have a little woogginess in their manufacturing. So we're going to have to account for that in how we do design and controls. And many years ago at Living Machines in 2016, we shared that we could use these approaches to fabricate robot scaffolds en masse using electrocompaction, and that the alignment of the collagen could be used to align muscle cells that are seated on the sheet. And this alignment was dependent on the alignment of the collagen molecules. Some of you who were here back in 2016 may remember this robot, which I usually say is moving, but it's not actually moving currently. Well, video has frozen, so to not anger the PowerPoint gods, we will not try to get it to play. It basically looks the same because if you remember this video from before, this robot is exceptionally slow. So while we showed we could use this material to build robots that were capable of locomotion, we saw a number of issues. For example, we had kind of limited deflection of the robot legs and low muscle forces from the monolayer culture of muscle cells that we seated on top of this, right? And this really gets at that problem of well understood mechanical properties, right? We needed a better way to kind of tune the mechanical properties of this material so that we could design the robot to match the muscle forces that we were going to get. Well, when I first started at CMU back in 2018, we began looking at this problem again. And we asked ourselves whether we could create ELAC threads and sheets with material properties more amenable to use in robotics. And this led to the question, how do ELAC mechanical properties vary with manufacturing parameters? There are several knobs that we can tune during fabrication, including the compaction time, collagen concentration, and compaction voltage. And my PhD student, Wenhan Sun, designed a series of fractional factorial experiments to investigate both the individual effects of these parameters and any joint interactions, and looked at how that affected the mechanical and geometric properties of these materials. For the full study results, you can check out our paper in the Journal of Biomechanical Engineering. But some of the key takeaways were that ELAC fibers with longer compaction duration showed significantly higher tensile strength, failure strain, and a larger cross-sectional area. And ELAC fibers made with low compaction voltage exhibited significantly lower tensile strength, modulus, and a smaller cross-sectional area. And joint effects of these manufacturing parameters could be used to expand the property range that we could get out of the resulting threads. So this lets us really tune these mechanical properties. And Wen went a step further and actually created a deep neural network-based recommendation tool that he trained so that you could put in the mechanical properties you wanted, and it would recommend what fabrication parameters to use. And so this moves us a lot closer to having well understood and reproducible mechanical properties in these materials. So what about that last one, right? How can we integrate this into manufacturing systems, right? I think this is going to vary a little bit between research groups and applications, right? Because the way that we manufacture our robots is really dependent on our ultimate goal, right? In my group, one of the areas we're really interested in is how can we have robust 3D muscle tissues for biohybrid robots, right? The video I showed earlier was kind of this 2D sheet-based robot. This was common in the early days of the field, where a lot of the robots were kind of cantilever-based. But recently, we've seen a trend moving biohybrid robots away from these systems and into three-dimensional systems. These are typically fabricated via casting or more recently via bioprinting. However, you may notice from these examples that all of these devices are relatively small scale or attached to soft or very lightweight structures. And I can only speculate, but I suspect this is partly due to the part of the research that very rarely ends up in papers, and that is these high failure rates in working with these very soft muscle tissues because of stress concentrations that can arise between the very soft muscle and the stiffer robotic structure. So when my group looked at how we could integrate our collagen-based biomaterial into manufacturing systems for soft biohybrid robots, we really wanted a manufacturing system that had the ability to print muscle tissue, and we wanted to integrate the ability to reinforce that muscle tissue with our collagen biomaterial. For this, we're collaborating with Dr. Adam Feinberg in BME here at CMU to integrate fiber reinforcement with collagen into his freeform reversible embedding of suspended hydrogel, or Fresh Prints method. Love the name. And so in this process, you can print very soft hydrogels, including extracellular matrix protein-based hydrogels, into a support bath, thereby allowing overhangs and really small features that would otherwise be unprintable. This technique is compatible with a wide range of bioinks that are of interest in biohybrid robotics, but also other hydrogels and even elastomers can be used with this approach. However, the resulting printed materials when you use ECM hydrogels are very soft and fragile, just like 3D muscle tissue. So we wanted to integrate our fiber reinforcement into this manufacturing process. To do this, we developed a new fiber reinforcing printhead that allows prefabricated fibers to be embedded in the hydrogel during printing. We combined this custom fiber extruder with a Replistruder 3 syringe pump and an open source 3D printer to create a multi-material fabrication platform. The fibers are guided by two guiding tubes that direct the thread through a pair of soft silicone wheels for extrusion. In the process, we begin by printing a layer of hydrogel into the support bath. We can print fibers embedded in that layer based on kind of layered patterning, or embed them in 3D throughout the whole hydrogel. An additional hydrogel can be printed as needed to encapsulate the fiber. The support bath can then be liquefied for part retrieval. Of course, this process is basically pushing a rope into jelly. And so the printability of any given fiber is going to depend on the fiber and hydrogel properties. This is why it's so important to be able to consistently fabricate these fibers with known mechanical properties. If the fiber is too compliant or the gel too stiff, the fiber will buckle. So we developed both analytical and computational models for the printing process to allow us to provide a printability model, and validated it experimentally across a number of fiber sizes and types. When fibers within that green zone are printed, they can be fully encapsulated within the hydrogel. And our initial prints demonstrated sub-millimeter deviations from the print path. This print fidelity can be further improved using closed-loop computer vision-based control of the printing process. Additionally, we can use chemical cross-linking to improve the fiber gel bonding, which we've tested through a series of pull-out tests, which found that while the initial pull-out force required was quite low, we could increase this tremendously with cross-linking. And as our original goal of this project was to reinforce hydrogels with this fibers, we also performed tensile testing of fiber hydrogel composites, and we found that the fiber reinforcement significantly increased the ultimate tensile strength and tensile modulus for the composites. As one would expect, the LFE Fresh also improves composite strength and bending. This approach is, I think, broadly extensible even beyond biohybrid robots to anywhere where soft, reinforced composites with renewable materials are desired. Our process is compatible with a wide range of threads and hydrogels. We've tested it with both alginate and collagen hydrogels, and with threads including single ply and multi-ply, polyester, silk, nylon, very tiny metal wires, of course, collagen, and even some custom threads, like this one in C, which is alginate with embedded silver nanoparticles. It can be used to print single or multi-thread patterns, and can even print structural reinforcement in 3D. And so with this, we've got integration into the manufacturing system. And moving forward, we're working on now printing this along with muscle. So with whatever time I have remaining, I do want to spend some time thinking about the challenges and future directions for the field of biohybrid and renewable robotics. Admittedly, there are many challenges that I think face the field right now. We must address challenges in longevity, right? If our robots are biodegradable, how do we ensure that they can achieve their mission objectives before they degrade? We need to be able to scale these approaches, right? Right now, our biomaterial fabrication is fairly tedious and manual, right? For broader adoption, we need to be able to manufacture at scale. The use of bio and living materials also introduces new controls challenges, right? There's a degree of stochasticity in these systems that we're going to have to account for with new control systems. And we're going to need to expand many of our approaches to additional materials. And when we've got some kind of roadmaps now for how we can do that. And of course, this is all going to need to come together with design tools. In the short term, this means we need solutions for biocompatible packaging, multifunctional soft structures, and scalable manufacturing. We need to continue to improve force capabilities in bioactuators and add integrated life support systems. Building on that, we can start to think about integrating living neural control circuits into these systems to reduce the need for electronic components. And we can bring this all together with manufacturing and design tools. So what can we as a community do to address these challenges? In thinking about this, I really came back to that biohybrid search that I showed earlier. And in looking at the Web of Science categories for papers being published in biohybrid research, we see a tremendous range of disciplines and multidisciplinary journals that are kind of highlighted in this space. I think this really emphasizes that biohybrid research is going to require a diverse STEM workforce, right? We need to bring together all of these researchers from these disparate backgrounds. We can really all strive to foster that multidisciplinarity and diversity. I've been incredibly fortunate to have the support of this community and many fantastic mentors during my training. As a PhD student, although I was officially in the lab of Dr. Roger Quinn at Case Western Reserve University, I had this opportunity at the transition between my master's and my PhD to start thinking about muscle on robots. And so during my PhD, I was co-advised primarily by Dr. Roger Quinn, an expert in bio-inspired robotics, Dr. Hillel Cheel, who we heard from earlier this week, who is an excellent expert in neuroscience, and Dr. Ozan Akas, who is an expert in tendon tissue engineering, right? These are very different disciplines, but these three mentors encouraged me to cross multidisciplinary boundaries, as well as the boundaries between their labs, right? I was able to be trained by grad students and postdocs in each of these disciplines, and I was encouraged to receive that training. I think this support is critical in shaping the kind of multidisciplinary academic path that I've been able to pursue, and is helping shape how I build my own lab. As PIs, there is so much we can do to foster this type of training in our students and trainees. And this is important because the very heart of our discipline, bio-inspired, is going to build on our own worldviews, right? How we are inspired by animal systems is a direct effect of our lived experiences, right? This diversity in lived experiences is going to be critical to stepping outside the bounds of traditional disciplines. And diversity can come in many forms, right? It could be from education in multiple disciplines and building diverse backgrounds in your research group, right? But it can come from hobbies, right? And what people like to do in their spare time. It can come from any myriad of lived experiences that shape how you view the world, right? So to the trainees in the audience, I really want you to think about how to question and explore the traditional disciplinary boundaries in your field, right? For those of us in more applied sciences and engineering, how can we innovate and advance these multidisciplinary spaces to bring them to broader applications? And to the PIs, just as I've been lucky to receive this in my training, please continue to inspire and support this type of multidisciplinary research for your own trainees. So where is biohybrid robotics going next, right? I think most immediately, there's a lot of great research to be done in novel materials integration. And this materials integration is really going to allow us to bring biohybrid robots into the field as models for biology, as tools for health and medicine, and as tools for environmental monitoring, without the introduction of potentially hazardous and non-renewable materials into these ecosystems. So I think I'm probably nearing the end of my time. And so I really wanted to leave everyone with a question today of how can you leverage renewable and biodegradable materials in your own research? I've shared some slides with results from my own group today. And so I always want to take a moment to acknowledge the people who actually do the work in my lab. We've got a great team here at the CMU Borg. And of course, we've got collaborators all over the country and the world, both at CMU and externally, who've really made this work possible, as long as our funding agencies. And I hope that we have some time for questions and discussion. Thank you.
Lovely. Thank you, Vicki.
Yes, thank you.
Roger,
And
there's a
looks like we
question.
have a question from Roger, yes.
Vicki, absolutely wonderful presentation. Thank you very much. And I probably agree with everything. I say probably just because I have to hedge, but I absolutely fantastic. If I was to challenge you on anything, it would be a very slight challenge, and that's just that just to just to feel someone always has to ask these questions. So okay, when living tissue dies, there is outgassing, there are contaminants. That's why we bury dead things, right? Also, when living tissue, if you actually want to live long enough, you want to make a robot that actually lasts for years, it's going to have to take in nutrients, and it's going to have to excrete also. So we pick on cows. There's like, I just looked it up, there's 1.5 billion cows in the world, and they outgas, they excrete, and there's one of the problems. But there's also almost 8 billion people in the world who also do those things. So this is a problem we have. This is one of our major problems, right? So now we're talking about making robots. They'll do the same thing. So you have to, but at the same time, because someone can say, oh, we're getting really good at recycling aluminum and steel and glass and various other things, and hopefully we'll get good at plastics. But anyway, so that's my challenge.
Absolutely. And I think I'm very fortunate. I'm very early career. And so I can think crazy long-term thoughts, right? And certainly we don't have the solution to that right now, right? We don't even have the solutions yet to really get these things beyond the petri dish, except in the case of cyborgs, right? And so this is going to be one of those basic research questions that researchers getting into this field are going to have to address, right? I think there's a lot of possible avenues to think about this, and it's going to require researchers from multiple disciplines, right? Before we even consider deploying these in the real world, we need to work with ecologists to really study the type of impact they might have, right? The other challenge you didn't raise is if I introduce an edible robot into the environment, is that going to mess up the food chain, right? I don't know. I need ecologist friends, right? So, maybe there are possible avenues to address this through symbiotic multi-organism systems, right? We've seen in biohybrid robotics a lot of great genetic engineering of bacteria, right? Is this, crazy sci-fi off the top of my head thought, could we genetically engineer a gut microbiome for these biohybrid robots? I don't think that's outside the realm of possibility for the field. And there's probably also some in-betweens, right? Which is why I think this is relevant to people even outside of biohybrid robotics, right? As an in-between, can we use these renewable structural materials, biodegradable materials, as things like the chassis of the robots, right? Kind of reduce the amount of non-renewable material we need in traditional robots, even without integrating muscle or neurons, right? But these are very real challenges that we don't have the answers to.
Good answers, thanks.
Yeah, go ahead, Will.
Fantastic talk, Vicky. Your work always prompts many internal discussions and exciting thoughts about where things can go. My question is just kind of a general question about the state of the field, which is that we're talking about a lot of building things using biomaterials. I guess my question is, what is the current state in the field regarding building things harvesting existing organic material versus growing organic material from some set of starter cells? And kind of going along with that, not to be political anyway, but we're seeing in at least the United States, the effects of different shifts in public perception and legal authoritative perception of different aspects of different things. And so I could see a potential problem going forward, especially in lines along things like the cyborg type research and things that are harvesting existing organic material. So it's kind of a weird, kind of nebulous question and direction, but I guess what are your nebulous thoughts on my nebulous question?
Awesome. Yeah, this is a really important question. As far as the state of the field goes, I think it varies based on kind of which of those three categories of biohybrid robotics that researchers are working in, right? Most of the biohybrid robots with individual muscles that are kind of on synthetic scaffolds, most of those are currently being fabricated using tissue engineering techniques, right? So we're growing those up from the cell level. And there's a lot of basic fundamental hypothesis-driven research that needs to go on there to really understand that process and be able to translate that further, right? In the cyborg community, of course, they're going straight to the using existing materials, right? The existing organism. So it really does vary by kind of sub-discipline. It's a small field to have sub-disciplines, but we do. To the question of ethics and government regulation and all of that, this has been something that, many of us in the field, you read our papers, there's almost always an ethics statement at the end of the paper, because we're very aware of this, right? We exist in kind of an in-between between robotics and animal research. And many of us kind of try to trend towards reporting this like animal researchers because we are working with living materials, right? But this is something that has not really yet been formally addressed in the literature. Recently, there have been a couple papers. Nicole Schu's got a really great paper of the ethics of the cyborg jellyfish, that if you're interested in this, I think her and Dr. John DeBeery are on there. It's a great paper. And for anyone who's attending Mars next week in Toronto, Rafa Mestre, who's presented at this conference before, is going to be giving a great talk on some work that we've been doing on this problem of ethics and policy, actually bringing in ethicists, right? Because I think that's an important step, is to not just decide these as robotics researchers, but really bring in experts in ethics, bioethics, and policy. That's a nebulous non-answer to your nebulous question, but it's something we're working on.
Thank you. The best kind of answer to a nebulous non-question.
Ken, do you have a question?
Yes. First of all, awesome talk. It definitely got me thinking about a number of things, actually many of which I've been thinking about lately. So I have two things that I kind of wanted to bring up. One, I was thinking about our a lot about our carbon footprint and kind of what we're introducing into our system, and I was into our environment, and I was wondering if on our most basic level, basically, what we're trying to do without producing too much waste is like, are we really just trying to reduce the amount of entropy, second law of thermodynamics, that we introduce into the system? Like, is there a way to just kind of assess, like, whether it's waste through a biohybrid system or a biologically like inspired robot or a biohybrid robot, or a cow or people or electronic waste? Like, are we just trying to simply reduce the amount of entropy that we put into the system? And then my second part, which is very different, I guess, is to tie back into Hillel's talk, like, where do we draw the line between like what is living and what is non-living and what we're comfortable using as a robot? Like, we don't want humans to go into these mines. We don't want trained dogs. We don't want trained rats. We don't want, so like, as you blur this line between artificial and biological, where do we feel comfortable, like, saying, okay, here's this biohybrid robot that we're okay with morally putting into the field, a hazardous field or something? So thanks.
Yeah, I'm going to address this second question first. I think that is going to be really interesting to figure out, right? As especially the cyborgs are really getting there, right? They're getting to where they could be deployed in the field. Dr. Hirotako Seto has some really interesting search and rescue applications for cyborg beetles, right? And they can control them by a human operator from outside of a rubble pile, right? So those are potentially headed out into the real world. This is where having a breadth of diversity of researchers is also going to be very important, because I think some of those lines that you ask about are probably culturally dependent, right? And we have to think about this as a global research institution, right? It's certainly not something I'm an expert in. And so that's why, you know, I really think we have to bring in policy and ethics experts from these different disciplines and areas. You know, in my own work, right, we don't build any robots that can reproduce themselves, right? So by some of the definitions we talked about that Dr. Chill talked about earlier in the week, are they alive, right? Well, the individual cells are definitely alive because they can divide and grow and expand and all of that, but the robot can't, right? And the robot is entirely dependent on us for sustenance. Although to Roger's question, we're going to have to get over that somehow to get them out into the field, right? But, you know, there's been the recent Xenobots work, right? Where the kind of motion of this little Xenobot robot fabricated from cells from frog embryos was able to push stem cells together into balls that then self-assembled and then themselves could go move in the environment, right? Is that alive? Possibly. The motion's not super directed yet, right? And they just have these ciliated cells driving them. But I can answer your question with a question, right? Like, what can we as a community do to try to address this before it's a problem, right? We certainly want to get ahead of concerns that the general public may have, right? Because we want this research to ultimately benefit people and benefit our global ecosystem, right? So I think now is a really great time to start having these conversations and bring the research field together to think about them. But it can't be something that I stand here today and say, this is what we're doing, right? We have to bring together as many of the relevant stakeholders as we can. And I've lost your first question.
Well, that was a great response to that. And yeah, the first question was like, as far as reducing like e-waste and just waste also in general, like on our most basic level, are we just trying to reduce the amount of entropy we're creating?
I haven't thought about it that way, but that is very interesting.
I guess I just feel like, you know, is there a way to simplify the problem to the extent that, well, maybe this complicates the problem, but like we're constantly trying to not make an impact on the environment, but also not make an impact on the environment. And like from an energy standpoint, we're trying to conserve, you know, everybody talks about renewables and we're trying to conserve. And so like, you know, we're not trying to heat up the universe. We're not trying to introduce entropy into the system too quickly, basically.
Mm-hmm. Yeah.
I don't know.
We probably need to start thinking about the energetics of these robots. And, you know, I remember years ago in grad school, this was also like always a debate about like, how do you calculate the cost of transport, right? Well, do you count charging the batteries, right? Or do you just measure after the batteries? Which losses do you account for? All of that. So we probably need to map out a standard as a field for how we quantify energy use of these emissions, right? Maybe after, as they're decaying, like Roger said, and environmental impact, and have actual standardized metrics that we can report so we can actually compare between different research endeavors.
I completely agree. I think that's a absolute great idea.
So we need some people who do research in energy. If anyone's out there, DM me. We've got a Discord.
Excellent. Thank you again, Vicky. And let us take a 15-minute break and come back for our first talk session. We'll actually have some time in the middle of this session as well to potentially discuss these things more. So yeah, we have several discussion times throughout the day. So see everybody again soon.
Cool. Thanks, Alex.
Hello, everyone, and welcome back. We have a slight change to our first session schedule. Unfortunately, our second speaker has woken up very ill and is not able to give their talk today. So we'll have a discussion session. I think we'll carry forward some of this discussion of what is living and ethics surrounding that. Partway through this session. But before that, we have a talk on integrating spiking neural networks and deep learning algorithms on neurorobotics platform. First author is Rachel Stenterford, but I believe we have Thomas Knowles registered for the conference.
Yeah, but Rachel's now left the lab, so I'm just standing in for her.
Okay.
Cool. Am I okay to start sharing?
Yes.
Cool. Right, can you all see what you should be able to see and hear me properly, etc.?
I believe so. You see your slides.
Lovely. Should I just start now or should I wait?
Yeah. Start now.
Cool. All right, so yeah, thanks very much for having me. So yeah, as said, I'm talking about how we've integrated spiking neural networks and regular deep learning networks on the neurorobotics platform. So this is a collaboration between the group I'm part of at the BRL. So that's Rachel, myself, and Martin, and that was supported by the group at TUM, which is Benedict, Denise, and Fabrice. Also, a big shout out to the HPP itself, which has funded this work, and also, well, my entire scientific career to this date has been funded by the HPP, so big thank you to them. Now, just as by way of introduction, just to sort of situate what we've done within the rest of the field, rest of the conference. So we're kind of on the sort of backward stroke of neurorobotics, trying to use robots as a platform to explore various questions in neuroscience, particularly looking at navigation, multisensory integration, multisensory sensing, and predictive coding. And this particular work is the culmination of several other threads of prior work that we've done, all surrounding the mammalian head direction system. And this work is bringing those various different threads together on the neurorobotics platform to try to find sort of new research directions and new insights. Now, due to the nature of this, I will be touching on a lot of these threads quite lightly. So, as always, just feel free to ask questions at the end if you want me to expand on any particular area. So the first component is this nest model of the rodent head direction system. Can you see my cursor, by the way? Cool. I'll just change to a spotlight. Is that hopefully a bit easier? You see the red dots showing up here?
Yeah.
Yeah, lovely. So we've taken the head direction signal to be generated between the lateral mammillary nuclei and dorsal tegmental nuclei. These two areas have like a very strong reciprocal connection. And as per the SCAGS model, we've taken this to be a ring attractor network. So the DTN here, which is made of excitatory cells, that's connecting across to its neighbors in the LMN, which is made up of projects inhibitory connections back. So the DTN will project across to the LMN in a sort of a kind of a Gaussian connection weight going all the way around the ring. And the LMN will project back to all but its neighbor. And so what that ends up giving you is a winner-takes-all network where whichever the most active cell is has this sort of Gaussian bump of activity, of spiking activity around it. And then you can feed in information regarding the animal's head direction to then knock that bump either clockwise or anti-clockwise around the ring. But as you can see from the figure at the top, if you plot this spiking neural estimate it will drift over time due to various integration errors. So it'll sort of wobble around the truth for a while, and then it'll start to deviate off, and eventually you've got a very inaccurate measure of head direction. So in order to correct that, there we go. So in order to correct that, we bring in a predictive coding network, which is written in TensorFlow called MultiPredNet. And sort of we're taking this to be part of Cortex that is supporting sort of navigation in the hippocampus and adjacent areas. So the way MultiPredNet works in brief, and again, apologies for not having enough time to go into this in more depth, but the predictive coding network is always trying to predict the activity of its lower layers. So here you'll have the sensory streams that it's looking at. You'll have various input and hidden layers that are looking at the activity from these layers. And the whole time through this, it's trying to predict the lower layers activity and just get better and better at predicting the incoming data as it goes forward. What happens with the multimodal latent layer is this layer is trying to predict both incoming data from both streams. So over time, once you train this model, you end up with a system that not only is making accurate predictions for both its modalities, or well, two or more, as many modalities as you want, you can plug into these layers. It's predicting both visual and head direction input, and therefore, if you were to knock out one of these streams, so for example, if you knocked out the head direction information, it can use, because it's, again, these predictions are inherently multimodal that it's trained to, it can, based on the remaining visual data, predict the head direction associated with it. So, you know, if it sees a particular sort of landmark tower in the distance, it's associated that with, 270 degrees. It can then, if you knock out the head direction information, it can predict that information. And we, like I said, we wrote this in TensorFlow, and we installed this alongside the NRP and linked it together via transfer functions. So as for the robot we're using, so is the video playing okay? Can you all see it moving and whisking? Awesome. So this is Whiskeye. As you can see, it's got a lot going on. It's got a nice, multi-jointed neck and head. It's got, a variety of different controllers that can make it do some very interesting behavior. So here it's, investigating with these lovely whisker arrays, the hand that's interacting with it. And it's been criminally underutilized in this project because we haven't used any of the whiskers. We've just used the cameras and the odometry. But the main reason we stuck with Whiskeye is because it gives us some congruity with all our prior research. And once again, I invite you to look at our other papers to see where we've used these capabilities a bit more in depth, or the full breadth of the capabilities. So now we come to the NRP itself. So again, as you can see, and as was in the introduction slide, the NRP lets us host all these components kind of in one sort of centralized system where we can have them all interact together. So in this window here, you can see you've got your gazebo environment with the robot that we've made an STL model of it, imported it in, attached the controllers to it, and now it can wander around this virtual environment and get a lovely, rich visual scene. We've also got this neural readout, so we can connect to Nest, run Nest models on it and connect to Nest and see the readout. And we've also got this sort of live coding environment where you can change parameters of the Nest model and the transfer functions. The transfer functions being the sort of decorated Python code that links the various components together. And keeping this all in sync is something called the closed loop engine, which is a little bit like the operating system of the NRP. It just makes sure all the components are hosted correctly and makes sure that all the components are running in sync. So nothing's ever working with stale data. If there's any slow components, they're allowed to finish their particular computation cycle, and then it moves on to the next component. So everything is communicating in lockstep. So this is what we ended up with. We've got this, the model of Whiskeye here that can see a nice rich visual scene. We've got the Nest model that's receiving vestibular information, or rather odometry information from the robot, that's adjusting its estimate of head direction internally. And then sitting in the middle, you've got this sort of machine learning black box, which in this case is MultiPredNet, but in prior work, we've done other machine learning methods as well for point of comparison. And this is outputting predictions of what the robot will be seeing and what the head direction should be at that particular point. And by outputting things in the same or a very similar format to what the neural representation of head direction is, we can then inject this as current and adjust the position of the bump on this ring attractor. What this does is, whereas previously we had this neural representation drifting quite a lot, or the representation of head direction drifting quite a lot, if you apply corrections, so first of all, train the model on the ground truth odometry, and then output those predictions and inject them in as current, you can keep the bump constrained to the proper head direction. And as you can see, these the estimate keeps quite nicely aligned with the robot odometry. Then as a further step, of course, an animal doesn't actually have access to its own sort of odometry. It doesn't have a perfect compass with it at all times to tell it which direction it's facing. And so what we used instead was this initial window of, it's not drift free, but it's got very minimal drift. The integration errors haven't had time to accumulate. And we use that information as training data for the system. And then we showed that by applying the corrections from the network trained on its internally generated estimate of head direction before it starts to drift, then we're able to apply good corrections. And it's not perfect. It's not quite as good as the ground truth, but it's still close enough to keep the two estimates in register. And then lastly, I hope this GIF is playing for everyone in the bottom right. Awesome. So with the NRP, we get this nice live feedback with the two systems interacting. If you run them in batches offline to each other, then obviously you don't get this nice interaction. So we wanted to look at to see what the sort of predictive error signal comes back from the predictive coding network and how that changes in a occlusion experiment. So you can see Wisky up here rotating, and we just basically made it so that whenever it wanders into this segment, so whenever its head direction is in one of these angles, denoted by the red area, then it would its cameras would just completely switch off. So it has no visual data when it's looking in these particular directions. And as you can see, as the prediction in blue from the multi-prednet moves through this, it's going very well. It's tracking the ground truth. It's agreeing with the internal idiothetic estimate. But when it hits this dropout zone, it completely collapses and it has no idea how to relate that, the non-information from the visual scene to head direction. So it's become quite dependent on the visual information. But then once the information comes back, in the meantime, the idiothetic estimate can continue tracking head direction. It doesn't drift that far in that small window, and then it recovers back to continue predicting as before. And the thing that we're quite excited about with this error signal is we think this could be a prompt for future work, looking at a way to make this sign of uncertainty in this error to indicate certain behavior or the need for certain behavioral changes in the robot. So for example, as this error, if you have like a transient error like we're showing here, where for a certain segment of the environment, the error gets very high and then goes back to normal, that probably indicates that it's not that you're in a new environment, it's just that there's some sort of occlusion. There's maybe an object in your visual field that you're not, that's blocking your view of the horizon. So that could then potentially trigger some sort of transient error, would potentially trigger maybe an obstacle avoidance behavior, so it can then recover its more accurate estimate of head direction. Or alternatively, if it reaches this plateau and just continues to plateau along, that probably indicates that it's moved into an environment where the context is completely changed, or the mapping between head direction and the visual scene is completely changed. And that could be a trigger for relearning this association. Again, that's the dream. That's the potential for future work. We're still hoping to look into that. Right. So just to summarize, we've got a model that's again got some biological relevance, particularly with the DTM LMN model. You can sort of analogize some of the other areas loosely. So vestibular from the supragenial nucleus, and the multisensory, we're saying loosely, could be maybe something like the post-rhinal cortex feeding into the navigation system. We've shown that you can get a teaching signal out of this ring attractor despite the fact that it drifts. And therefore, the machine learning and the spiking neural network systems can sort of bootstrap each other to create a system that's better than the sum of its parts. We've validated the use of the NRP as a sort of a good host for all this, and can bring all these different threads together and have them all interact, including bringing in TensorFlow on a transfer function. And it's pointed us in the direction of future work. So again, I've gone over a lot of this before, but so basically the error signal could be used to trigger either behavior or relearning, or sorry, remapping, sorry, learning. And the again, the hope in the future, so far we've used a pre-trained predictive coding network that we trained offline and then imported in. We'd be hoping to use a sort of fully online, trained from scratch, end-to-end learning system in the future. Right, that's everything. So yeah, thanks very much for your time and attention, and happy to take any questions.
Excellent. Thank you. Do we have any questions from the audience? People are getting their computers. I definitely have some questions. So I guess some things that kind of has me thinking is that like the visual environment like that you showed would definitely, like as the robot moved, like the kind of landmarks would move generally as well. Is there a way to try to account for that?
So I mean, the way we've done it so far is we're purely using distal information. So if I just have to quickly take you back to that, you can sort of see it here. So all this, we've done some tests with occluding it with things like these columns here, for example. That does cause some interference, but it seems to entrain mostly to the fairly static backgrounds. So for the most part, that isn't really an issue for it. Obviously, in a more natural environment, particularly for a rat, where there's lots of tunnels and branches and things. Yeah, we'd have to think of a way to either get it to try and ignore that, or to change your navigation strategy depending on which sort of local environment you're in. So if you can see to the horizon, then obviously you can use this sort of method. If you're you're really enclosed, then obviously you'd have to use something different. Maybe that's where the dead reckoning, the integration would come in.
I guess I have another question about the training aspect
Go
as
for it.
well. So you like divided it into this like specific training data time and just kind of implementation. And my general understanding with the biological systems is that it would all be much more kind of constant and loosey-goosey, where you're probably constantly updating your reference. Is there a way to maybe kind of mix that in a little bit more seamlessly?
Yeah, so the multi-prednet has some of this. So oh, go away, West guy, come on. There we go. So there is a kind of the way the way the networks works is it's got a sort of state at each layer that it constantly kind of refines based on how the incoming sensory data changes, which is why you see that prediction collapse very rapidly when the visual data goes. So there's a little bit of that adaptability in there, but you are right, that does again rely on there being the pre-trained weights between the layers that has that mapping sort of in there intrinsically. Yeah, we haven't tried, well, we've tried briefly. We haven't had any massive success training this kind of network online. Probably partly because that was never one of its sort of design criteria when it was put together. Yeah, we've had a couple of thoughts. I think ultimately it might come down to there's another group that Cyril Penartz is part of, who's been trying to do a spiking version of this not necessarily multi-prednet, but some spiking predictive coding, which I think would possibly suit that kind of live interaction a lot better if that could be made to work. So yeah, I'm afraid that's all I'd have on that.
Great. I think we have time for a question from Will.
Hi, great talk. I have a two-part question, not about your visual and kind of state estimation network, but kind of questions about the current state of the neurobotics platform as a whole. Is that okay, or would you prefer questions?
Go for it. I'm not a developer on it, but I'll use my best understanding. I'll be as helpful as I can.
Okay. So my two questions are, so Neurobotics Platform does a great job of integrating so many different things together into one tool, right? The drawback with that, of course, is total speed, right? When you're interacting with so many different elements kind of together. And one of the common issues that's coming up in applications of robotic simulation is getting things to simulate faster and faster, to do more iterations, to kind of, I guess, to kind of validate the use of simulation in design. So I guess one question is, how is Neurorobotics Platform kind of looking to accelerate how fast things can be simulated compared to real time? I get that that's a very kind of Neurorobotics Platform developer question. So if you're not able to answer that, that's okay. And yeah, and then
I'd say briefly on that. I mean, it's very much, it's a bit of a cop out, but it's kind of so I was with a HPP meeting last week, and one of the developers was talking there, and they did say that this is something they're aware of. It's something they're trying to fix. So this is NRP 3.2 that I'm showing off here. With the NRP 4.0, they're completely overhauling the architecture and making it a lot more I don't know if it's async, containerized, and coming up with sort of a nice common interface to put all these components together. So hopefully in the future that will lead to some sort of speed up. It's something they're definitely looking into. Yeah.
Thank you. And then just really briefly, the last question is, how are things going with, because I know there's been a lot of work in designing these networks in the NRP and validating their success within the NRP. Has there been, how is the state of transferring networks that, like in this example, run on WSCI in the simulation? Has there been any work in transferring these onto the real hardware and demonstrating success in hardware versus in silico?
What's using this like as a prototype and then moving that onto the physical system? So yeah. Yeah, I wish I could say, oh yeah, we've done exactly that, but no, we haven't done this in this case. I'm trying to think of an example for you from another group. Yeah, because we sort of started with the physical and then we moved on to simulation when the pandemic hit, and so we haven't really since it's wound down a bit. We haven't really gone back to the physical robot yet. I mean, hopefully in a year's time I can come back to you and say, yes, we've done this and it all works. As much as possible, this is all kept congruent. So like the control architecture was all translated into Python. It was run on the physical robot, and then that was used to form the NRP's version of the controller. So it's kept quite congruent, but of course, obviously, with it being a simulator and then the NLP specific requirements, yes, there's probably some divergence there that we'd have to account for if we went back the other way.
Gotcha. Thank you very much. Great talk.
Thank you.
All right. Thank our speaker again. Thank you, Tom. And we will, I guess, move into a discussion session for about 15 minutes. I think maybe we should do breakout rooms if Ken can move us into separate rooms.
I can give it a shot.
Give that a try. Yeah, maybe somewhere between 5 and 7 people per room. And let's kind of discuss, what makes something living. And I would say also maybe diving into why do we care to define what is living?
All right, here it goes. Let's see.
Do insects feel pain? And now is that going to, that going to essentially really cause a problem for insect research and the cyborg things? And then it matters, right? If you, right, it's like, oh, it'd be much easier just that not worry about it. I can point out that I grew up on a small farm. I was literally the cowboy, and the feelings about animals were they don't feel pain the way we do. And I've always had a problem with that. As a kid, I thought about that, and it bothered me. I at this point, I'm happy to not be raising cows anymore. So it remains the problem. Ethics. What is life?
Yeah, no, I if I may chime in, I often think about that as well, and it's not an easy thing. I dabbled in vegetarianism for a little while not too long ago. I lasted about a month, maybe a month and a half. And because I watched one of these videos online, and it was like, oh, this is, it showed you some of the processes that go into it, and as a consumer, you just see that I love grilling out. I love the flavors and everything. but it is it is very hard, and to kind of like distance yourself, to separate yourself from like the beginning and the end, and 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 so like I wonder, you know, okay, with the advent of lab grown, you know, or ethically, what we consider ethically raised, or farm, you know, like will that sway, you know, how will that, I mean, clearly it's got some traction, it's got some, you know, there's some drive behind it, but will that kind of like alleviate some of these questions, or will it make it even more complicated?
I think for me, living through the 80s up to now has shown me that the longer I do this, the longer that I'm alive, the more conscientious we're all becoming. Right. So this list of things that that we consider in daily life just keeps growing. Right. And I am constantly adapting to a society that in most ways seems to be getting better. I mean, loudly regressing in some areas, but in general, I think that we're becoming better people and better scientists, right? And interestingly enough, I think that this collection of disciplines should be leading the path, right? And I think in every way that I can perceive, that's what we're actually trying. And I think that's pretty cool. It's neat to be a part of a group that's committed to it.
Yeah, when Ken said that he tried vegetarianism, I know people who are very healthy vegetarians. I know people who eat exactly the same things who are very unhealthy vegetarians, and it's DNA. You know, I looked into it. It comes down to our DNA. It depends on, you know, essentially where we're from, genetically, whether we can be healthy vegetarians or not. It'd be nice. I would like to be a healthy vegetarian. I'm not a candidate, apparently, based on my genetics. So Evan, you're young. What do you think of, did you watch Hillel's talk? Did you have an opportunity?
Yeah, it's interesting to think about the different degrees of sort of conscientiousness that we have towards life. Like, I know if there's a fly flying around in my room, I'd probably go to squish it. But if a cat showed up at my doorstep, you know, you don't squish a cat, you just don't do that. So it's interesting, like, where is that line of life that most people hold more valuable than others in terms of animals? Just interesting to think about that.
And one of the definitions of life is reproduction, and I can't reproduce. I suppose I could be cloned, but but then Hillel then corrected that. Well, I would say the newer thinking sort of solved that issue, I should say. But I would, you know, in terms of whether a robot is alive or not, it does. I would actually have. I used to be, there's some machines. Now I'm starting to feel differently about what alive means. It's interesting. My thoughts have advanced more toward more acceptance of what is alive. And I do catch spiders and things and throw them outside. If I can, I do. Sometimes I'm just angry and stomp on them, but often I throw them outside.
But you know, Roger, also in this discussion on what's life or not, it's also a little bit an artifact of how the brain operates, because we are always forced to impose categories on a very fuzzy reality. And also, I think in biology, you've seen this discussion about the definition of life going on for ages, and the biologists haven't sorted it out, because maybe it's just a very gradual transition, right? Why not? And then we could say, okay, then who cares? Because then it's a gradual scale, and then some things might be more alive than others. Some are at zero, okay, fine. Others are more hybrid or mixed mode, you know? So I think we should maybe not have too much of a hang up about it, because then, of course, you can also say, look, an artificial system can replicate or emulate some aspects of this multidimensional definition of life, but not all. And so what? Because there's certain biological systems that have a similar partial mapping to these dimensions, right? So would that not be a more constructive way forward, not have an almost religious debate, like, no, we have to have one border, and here's the US and there's Mexico, and that's it, right? Well, the nation state is a construct, you know? So how about that compromise?
That I like that. Yeah, avoiding the binary, avoiding the binary definition. It's kind of like how aware, right? I mean, the more aware, it's like comes down to the brain, right? If you're aware, humans are probably, we assume, the most aware of ourselves, of others, of the universe, or whatever, versus a spider, which is probably not. We can just look at the brain and see. It's clear.
Yeah. Well, but of course, we are the only species who has the capability to declare with a lot of self-confidence that we are very aware.
That's right.
Right.
It's our viewpoint. It's our viewpoint, right?
Exactly.
Yeah.
Maybe dolphins and whales have much bigger brains than we do, much bigger cortices, have also higher capacity for awareness. Why not?
Yeah.
Right? They control sort of the thalamocortical system. So,
Again. Yeah.
Hey, just thought I'd
Hello.
pop in and say hi.
Oh, yeah.
How's the talk going? Or the
Good.
discussion?
I think it's good. Yeah. 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 I guess I was saying, like, the renewable aspects are probably super important, but biodegradation would be for only specific instances. Biocompatibility as well.
You can have materials which are biocompatible, but they're not biodegradable or something like that. But it's quite difficult to see a robot like. Of course, we have to think about the life of a robot, and how we can use afterwards the different components in order to save. But yeah.
Yeah, but one question is really also important. Do we want to have living robots?
Yes, I don't think we need living robots. I don't think.
Because then we need also ethics and rights for living robots.
Yes, because so for example, I work with animals also, and we have so many regulations in Germany to work with animals.
Yes? Yeah.
And if you say a robot is alive, then he can feel pain, maybe, yeah. It can feel pain as this, then we need a different regulation for that robot. So we have to put it at the level of animals, at the level of humans, and in that point it will be.
Oh, hopefully not too many people were cut off in their conversations, but it is. I hope the conversations went well. You know, our group definitely had some interesting discussions. So it is time to move on to the next talk, however, and we've got a talk on robotic implementation of neurally based animal magnetic reception and navigation. So some more kind of navigation aspects, and from Andrew Harvey.
Thank you. Can you see my screen?
Yes.
Okay. Hello, my name is Andrew Harvey, and I work in the Quantitative Biology and Engineering Sciences lab at the University of North Carolina at Chapel Hill. And today I'll be presenting on the paper that I wrote with Dr. Brian Taylor titled A Real-World Implementation of Neurally Based Magnetic Reception and Navigation. So first, I'd like to go over a little bit of the background and motivation. Many organisms can sense the Earth's magnetic field and use that signal for orientation and navigation. Magnetoreception has been demonstrated in numerous species, including sea turtles, salmon, and birds. The mechanism through which organisms sense the magnetic field and the strategies they use to navigate are not well understood. It's hypothesized that there's no single organ that accomplishes magnetoreception, but instead that it may be accomplished by a distributed array of sensors within tissue inside the organism. We currently use GPS and other satellite systems for navigation, whereas organisms use local and natural senses. Systems like GPS are limited in that they're vulnerable to both intentional and natural compromise. They're unavailable underwater, and they're also expensive to deploy and maintain. We want engineered systems to be able to navigate autonomously without reliance on satellites or map data that must be continuously updated. And animals are examples of systems that can navigate autonomously without this outside input. From an engineering perspective, understanding how animals navigate with the magnetic field is potentially useful for the development of new navigation systems. Magnetoreception and navigation is also something that's difficult to study in animals, but modeling and simulation tools from engineering can help augment experimentation to explore these topics. The overall goal of this project is to use engineering tools to better understand biology and use biology principles to design better engineered systems. Earth's magnetic field provides a navigation signal with two components: intensity, or the magnitude of the field, and inclination, or the angle that the magnetic field vector makes relative to the horizontal. Drawing lines of constant inclination and intensity yields maps like the one here on the right. And intersections between lines of constant inclination and intensity can form unique or rare points that we refer to as magnetic signatures. And examples of some of these points are shown on the map as the circle and triangle. And one possible navigation strategy that animals may use involves using these magnetic signatures to move towards desirable locations or to avoid undesirable regions. It's not currently clear exactly how magnetoreception is processed by the nervous system for navigation. This study builds on previous work that developed a prospective navigation strategy that used magnetic signatures to navigate to a series of points while using neurally relevant sensing and processing. And that work was completed only in simulation, and this study expands on that by testing the navigation strategy with physical movement and sensing using real-world hardware. To test our navigation strategy, we created a simulated environment with a simplified magnetic field that doesn't represent any specific location on Earth, but does contain the same general features as the real-world magnetic field. We then placed 4 goal points in the environment and allowed an agent to migrate in a closed loop using magnetic signatures to test the strategy. In the real magnetic field, there are natural magnetic anomalies, and the intensity and inclination contour lines can be non-orthogonal and curved to various degrees at different locations. And the simulated field contains 2 contours also, one that represents inclination and one that represents intensity, and also contains artificial magnetic anomalies. In this model, a single anomaly at the origin. The contours can also be adjusted to be more rectilinear or curvilinear, and to be orthogonal or non-orthogonal at all locations. So in the environment, we can vary the curvilinearity, non-orthogonality, and also move the locations of the goal points. Our team has used this same environment in several previous studies to test navigation strategies before implementing them in more realistic environments. This navigation strategy incorporates a neural model for processing sensory input using a distributed array of virtual sensors. With this model, there are multiple pools of neurons arranged in a ring sensitive to a particular direction, and they're connected via inhibitory and excitatory weights, such that each pool excites nearby neighbors and suppresses more distant neighbors. And we feed the sensed magnetic field vector as input to each of the pools, and they essentially compete to determine which direction the agent should turn towards. To test the strategy with hardware, we used a magnetic coil system to generate magnetic fields. That's shown here in panel A. We also used a magnetometer to sense the magnetic field, and that's the top part of panel B here. And then we use a robotic turntable or rate table to rotate the magnetometer in the magnetic field, and that's shown here at the bottom of panel B. And for simplicity, we only used one magnetometer and fed the sensed magnetic field to all of the neural subpopulations, but this could be done with multiple separate magnetometers, and this is something we're currently working on to extend this work. Going over the process that the model uses during a simulation. For the simulation without physical actuation and sensing, first, we read the intensity and inclination of the simulated magnetic field at the agent's location. We add noise to that and feed it as input to each pool in the neural fields. Then we evolve the neural model to arrive at a desired turning direction. Then we rotate the agent in that direction, move the agent forward by an increment, and repeat the whole process. With this study, we added physical sensing on top of that process. Rather than allowing the agent to sense the magnetic field directly, we took that field, used it to generate a physical field using the magnetic coil system, and sensed that field using the magnetometer before passing it as input to the neural field. And for some trials, we kept the rate table fixed in place as the agent migrated. And for others, we rotated the table and magnetometer to match the agent's orientation at each time step before sampling the magnetic fields. And so here is a video of a trial in progress. So on the left side, you can see the rate table turning to match the orientation of the simulated agent. And then here on the top of that, there's the magnetometer also rotating inside the magnetic field. And then in the bottom right, this represents the inclination and intensity of the magnetic field, both the target and what's sensed by the magnetometer. And then over time, we can see the agent completing the migration and moving through the simulated space as it's sensing the magnetic field through the hardware and using its navigation strategy with the neural field to complete the migration. So these results were generated in the previous study that tested the navigation strategy using only simulations. And in all cases, the agent was able to successfully navigate to all of the goal points, but more extreme excursions were observed as the environment was made more curvilinear and more non-orthogonal. And these are the results that we obtained for the present study when integrating physical air into the setup. Trials were completed with 2 artificial environments, one that was more rectilinear and one that was more curvilinear and non-orthogonal. And 3 types of trials were completed: a software-only simulation that used the same methods as the previous study, a setup that incorporated physical sensing, physically sensing the magnetic field but not rotating the magnetometer, and then a setup that also incorporated rotating the magnetometer in the magnetic field. And as with the simulation-only results, the agent was able to successfully complete migrations for all of the trials that incorporated physical air. And the resulting trajectories are also very similar between the simulations with the artificial noise and the simulations with the real-world actuation and sensing noise. So in conclusion, the results of this study demonstrate that the combination of magnetic signatures and neural processing can in fact be used in a physical system as a viable navigation strategy. We demonstrated how a nervous system could potentially encode magnetic information for successful navigation, and that a nervous system with distributed sensors is capable of executing a strategy based on magnetic signatures. And this further supports the hypothesis that animals may use this strategy in practice. But we also provided an example of bio-inspired magnetoreception in an end-to-end manner, collecting physical field samples as input, processing those with a neural model, and rotating the robotic agent to complete navigation. And this system demonstrates processes that would be difficult to test experimentally in organisms, and also exhibits potentially interesting engineering benefits. For example, using distributed sensors, a system of this type may be less susceptible to failure due to one of the sensors being disrupted. In terms of topics that we'd like to explore in the future, we're currently working on expanding the system to include multiple magnetometers. The photo on the right shows the new setup that we're currently developing and testing. With multiple sensors, each subpopulation would receive readings that may be slightly different due to different air at each sensor. And we'd be better able to quantify how well the neural field performs when data for one of the subpopulations becomes corrupted or unavailable. We'd also like to run the system in a magnetic environment that resembles a real-world location, such as the North Atlantic Ocean. And in other studies, we've successfully demonstrated that this strategy without neural processing can run in real-world magnetic environments, but we'd like to test the neural model in those environments with physical sensing. We'd also like to add additional sensor modalities to the system so that we can fuse magnetic data with other sensory data such as visual, audio, or mechanoreceptive data. Finally, we'd like to compare results from our computational neuroscience-based method of processing to more traditional engineered approaches such as Kalman and particle filters to explore the similarities and differences between these methods and where they might diverge or could potentially be fused with one another. Thank you for listening. I'd be happy to answer any questions you have.
Excellent. Great talk, Andrew. Looks like we got Will up first and then Paul.
Andrew, great work. I have kind of just, it might be a dumb question, but I just have, what interests me about magnetic navigation, at least from an animal perspective, is that in my mind, kind of the classic examples of even animal kind of migration that use kind of magnetic signals to navigate tend to be either in small areas or, so just like fish swimming in a stream or something like that, or things that move a long distance, but just in one cardinal direction. So like butterflies migrating from the north to the south and back up, or like geese and that sort of thing. So I guess my question is, in a world where we have planes and things that move very fast across the world, how scalable is magnetic navigation? That's a very big question. So if you can't do a full answer to that, that's completely fine. It's just a general question.
Okay, I can try to answer some of that. So here's a depiction of sort of some of the lines of inclination and intensity in the Atlantic Ocean. And for example, migrations with sea turtles, some of what they believe is the turtles might follow lines of constant inclination to the west just to reach a coast with a specific inclination. So maybe that's one way that those types of animals use the magnetic field for long distance navigation. But what's nice about the magnetic field is it kind of forms a grid, and that grid covers the whole Earth. So if you travel at high speeds and we're traveling all across the Earth, it would be feasible to find unique magnetic signatures sort of all over the Earth to navigate to and sort of use those for long distance travel.
Very cool. Thank you.
Go for it, Paul.
Yeah, hi. So great talk. Thank you very much. And these must be also really challenging experiments, so I really appreciate it. But what I was curious about is robustness of the model that you present, right? So magnetic fields also deform. So can you say something about the robustness of your model to perturbation and also, let's say, long-term stability, and also how it would scale when you start to modulate the sensitivity of the sensors themselves?
Yes, I think this is something that we need to investigate further. So here are the results that we obtained with the physical setup. And so this is maybe sort of getting to what you were talking about with the sensors possibly having noise and then also having maybe difficulty in generating the magnetic field for simulations, or interference from outside systems, such as in our case, the rate table that we put inside the magnetic field generates its own magnetic field. And so that sort of disrupts our setup also. But so far in the testing that we've been doing, it has been robust to this influence, but we would probably want to investigate sort of maybe artificially reducing the quality of some of the signals, or the sensitivity of the receptors, to see how it responds to that.
Okay, and then do you believe the model itself that does the reconstruction, do you see this as a generic template that you will find in all animals that are sensitive to magnetic fields, or so it's like a generic solution that evolution found, or do you think it's just one of many?
I would think that it's probably one of many. I think, considering how navigation, what we know about magnetic navigation in animals, it seems like it's very diverse, and that different animals use different methods. And so this is just one possible method that they could use for navigation.
So if you could choose one animal whose behavior you would explain with your model, which animal would that be? You can pick one. You can pick one.
I have always been thinking of it in terms of sea turtles. That's sort of what we study a lot at UNC for magnetic navigation. And we sort of know that in order to stay in the North Atlantic, they don't want to go up higher into Arctic regions, and they don't want to fall out of sort of the North Atlantic region. So they may sort of avoid certain magnetic signatures, or be attracted to other magnetic signatures to complete their navigation. So that's sort of what we're trying to simulate with the clockwise navigation, is staying within the North Atlantic region.
Great. Well, great work again, Andrew. Thank you very much.
Thank you.
Thank you again, Andrew. If you have any more questions, you can take them into the Discord channel. And with that, we've got a short break, 15, 20 minutes. And when you come back, hopefully you'll be assigned to another breakout room for a discussion session. And the focus of this next discussion session is about jobs and careers. And it's really a chance for kind of younger participants to ask questions of older participants about career paths, jobs that they might be able to find, and how to kind of navigate the landscape. So, see everybody again in a little bit. everyone have opened up the breakout rooms, so feel free to hop into whatever room you've been assigned and start discussing.
Hello. anybody there? Hi, Nikki. How are you?
Good, and you?
Good, good. I was just checking in.
There's only
what do you think so far?
it's a great conference. It's very nice. It's very cool.
Good. Hello.
Hello.
How do you?
Nice to meet you.
Nice to meet you as well. Is it? How do you pronounce your name?
Goffredo.
Goffredo. Goffredo.
Yeah.
Hello.
It's a strange name.
Italian.
Yeah, but it's strange also for Italians.
Is it really?
Yeah, yeah.
Unique. Not strange, unique. It's special.
Yeah, thank you.
I saw your question, Goffredo. So yeah, you can just share your screen and take over the mic, and we'll once we're done with this discussion portion, we'll go back to the main conference room.
Okay.
Virtual conference room. And then you can, and then after, I think there's 2 talks before you. Are you the?
Yeah.
Okay.
Yeah, I saw the program, yes.
Okay,
Thanks.
okay. Yeah, sure, absolutely. Does anybody have any questions on jobs and careers?
I mean, for me, I'm kind of sad. I mean, I'm well, not sad, but like, yeah, I mean, it's been long since I finished my PhD, postdoc, so going towards the, well, not academic 100% direction, because I didn't want that. It's too much responsibility for my my personal taste. But yeah, no, I mean, I don't know if Goffredo.
Yeah, it's the same for me. I don't know if I will continue this path, let's say, because it's hard. Also, I don't know if you are, you know, the condition in Italy. It's been pretty tough.
Yeah.
About
I mean,
yeah. Tell me.
So what are you, PhD?
No, postdoc.
Postdoc, okay.
Yeah,
so
yeah.
you know.
Yeah, I know
Really?
very well.
Yeah, because you have like the marriage, which is the PhD, which is like, one thing that you have said, I do. So you want to make it work. So you you finish it, and then you go like, okay, what's next?
Next? Yeah.
And there you go like, okay, I could do a postdoc to see if I still like this relationship kind of situation. And typically it's not bad, but like I would say, and this is purely my take on this. I mean, unless you're very, very passionate about 2 specific things that I find that are important in academia, one of them is, I would say, teaching, because I think that kind of like is linked to having like and the other one is leading, because progressively my assumption would be that you go from postdoc, then you try to get some scholarships potentially, and then some grants so that you can go
manage
either as
also
lecturer,
your lab.
professor, and then making your own lab. Then if this is not like your cup of tea, then I think you have to find something that maybe can marry the things that you do, like an R&D. There are companies that do R&D, or go completely to industry or something along these lines, I guess. And the truth is that Europe, because I mean, I come from Greece, I was studying in Spain, now I'm in the UAE, I'm in Abu Dhabi.
Okay.
It's a very interesting place. I mean, salary-wise, it's not even comparable to the rest of Europe when it comes to this. I mean, if there is any interesting, like even for research, it's a lot better. Like now I'm a senior researcher here, so we do a bit of everything, a little bit of research and development. So it's a research institute, but like if it hadn't been for this offer, I would have I would have
skip.
I would have gone towards the industry because, you know, like, I mean, I'm 40. I'm thinking there's some time in my life that I start to need saving money, you know, like for the Vicky of 80 years old, I think this is important. But it's not, but then again, it's not that you cannot do this with academia. And unless you're really, really passionate, then you don't have like the microbe, the virus of that. But you can still do a lot of things in the industry, I have to say. Like, I have a lot of friends that are now, like I have two friends that I call them the power couple, because now they are in Novartis and they are like leading neuroscience and rehabilitation domains. And they're still doing some sort of leading and doing some sort of research, but they're working a lot with actual products and you know, it gets a little bit more managerial, the position, rather than research, but
their life is okay, let's say.
their life is set. Their life is set. I can tell you that 1 year, like, I mean, yeah.
Okay, thanks. It's the last drop to leave the academia for me.
I mean, you can, like, it's not that you can, it's just that, you know, unfortunately, there's my problem, I feel, because I see this also from other friends that they were in France and they were trying to get grants. There's so much competition when it comes to getting your own grants, and you have to have like also a lot of networking, which is also very important from what I noticed. Like, you know, you need to have affiliations or people that can support you, like research groups and big names that can support you. And like the competition, at least, for example, in France, is fierce. It's very, it's not that it's not doable. It's just that, you know, like, as long as you're not afraid of failure or rejection and you don't take it personally, then I think it's again, it's doable. But I don't want to influence or anything.
No, don't worry. The virus is still there since a long time.
That's good. And again, where I am doing research. Like, this is not out of the scope. So, I mean, so you can find like a marriage between the two and still do what you like, and also see if you can.
Yeah, but I guess most of us do research just because they are passionate people. Otherwise, I mean, it's the main driven point.
I would call it also a little bit of masochism, but in a good way, you know, like, you know, like you suffer, but you enjoy it, you know, like, I mean, you fail, you still enjoy it, you know, you have so many unanswered questions, but you still enjoy it because
I don't suggest you to come in Italy in any case to do research.
Honestly, I have collaborated with a lot. Actually, we are collaborating with a lot of Italian universities, and even in my previous lab, we were collaborating with IIT.
I'm from IIT.
This is TII, by the way.
Yeah. It's I can show you.
Where are you in that IIT?
IIT,
no.
Yes.
You're in IIT?
Yeah.
IIT is great. I mean, I've been working with Giorgio Metta.
I will speak with you separately.
In private. Yeah. I get you.
We won't share. We won't tell
I feel
anyone.
you. I mean, look, there's no place that is perfect, I assume.
No,
but like,
obviously.
University of Pisa. Then IIT that we have collaborated are very, very good. Then we also have a lot of companies that we collaborate that are doing, like Spin Italia, and some other companies that we collaborate that they also do this R&D. And it's honestly, I mean, the results that they produce also
No. But no, but in terms of results and in terms of also instrument facilities, I mean, it's great. It's the best place in Italy. It's a bit not comfortable for the career, let's say. Their policy is to be totally flexible, let's say, stay here for some very few short period and then go somewhere else. I mean, it's okay, but someone sometimes wants to grow their own career also in a place where they find out a good place to do research. I mean, I mean, we can speak separate.
I get it. I really do get it. But like, have you thought of scholarships like a Marie Curie? Because that at least gives you a lot of autonomy, right? And it's a very good thing for the PhD.
The problem then it's also to merge with your personal life. Let's say once you are in a relationship, you need also a person that is deeply in your own shoes, let's say, because
you're cheating on science.
I would like to have a safe life. I mean, you have to do your own. You have to choose sometimes. Life is a matter of sometimes you have to choose, and then I mean, research is beautiful, but or you have to sacrifice your life for science, or at a certain point you just say to yourself, I did something that I liked for a while, and then I decided to do something else. Let's say if you think like a normal job, I mean, everybody in the world does the same. Let's say they follow their own willing. So
and what's sad is that not everyone is doing what they like, right? So you've done that up to a point, I assume, by choice. So at least the good thing is that you get to enjoy what you do. So it's not 100% job, you know, like it's a like you might need to work weekends. I remember my PhD and my postdoc weekends were like commodity, like a little bit of a luxury to have, because you take it also personally. It's my work, it's my name, it's my thing, right? But then again, yeah, I mean, balance. For me, there was a point that I got a little bit burned out, and I noticed that like having a balance, it's extremely important when it comes to these things. Otherwise, you cannot give, not even to yourself, let alone to the thing that you like and you love, you know.
But you need to be balanced also in terms of what you are doing, because at the end we are sharing our knowledge with the world, let's say. So it's not our job. It's thanks to a multiple factor of stuff that we achieve this result. And then I mean, it's this romantic way of the science that sometimes we we miss or we lack. And then also for this reason, I told you, for example, this institution is very beautiful, but if you need just to showcase that you achieved a very beautiful result, but at the end the human capital is not that happy, because they would like to have they would like to continue their career that doing what they like, then I mean, maybe you missed something. Okay, maybe you have plenty of patents, plenty of very good paper, but
if you're lucky, yeah.
If you're lucky, yes.
Pattern is not something that comes so easily.
But But I mean, I don't know. It's a complex life.
It is, and it's like we have this in Greece advertisement that was like, you know, for like private colleges, which is like, after high school what? And I feel that every step of my academic career, it's been the same question. So After bachelor, what? Master. After master, what? PhD. After PhD, what?
Postdoc.
After postdoc, a second postdoc or something like that, you know, like it's always like, you know, places where you, when you finish that, you go like, okay, so now what?
Yeah.
When do I do adult stuff? Do I continue with this thing? What do I do?
Yeah.
Jack, what is your status? You're not
You're
heard.
on mute.
Or we don't hear you if you're not muted.
Probably don't hear me.
Oh,
Yeah,
there. Yeah.
Yeah. Is this the microphone? That is very cool. That's very pro. That's very.
You're still very quiet, Jack.
Oh, for Christ's sake, something's
There we
gone wrong.
go. That's a little bit better.
Let's just try and mute and remute. Right. So PhD student, and going back to what you were saying about what next, for me, that question was like February of 2020, because that was when I was doing a master's, and I was just sent an email saying, hey, from my bachelor's supervisor saying, hey, I've proposed this project idea, fancy doing it? And I said, yes, and this is where I am. Pretty much if you don't take the opportunity, you'd never get anywhere.
No, clearly.
I don't know how much that applies to the professional life. But yeah,
I think it applies.
We are leaving.
Did you also get this message that the breakout
Yeah,
rooms
yeah.
are
yes, yeah, that's basically we've got like a time limit of 20 minutes, and you had a
20 minutes passed already?
Yes. You had a lovely like 20 minutes, like 15 minutes.
Oh!
I was thrown in
so
about
passionate.
5 minutes into the question or so. And it's just like, when's the best time to come in and just wave and say hello?
Just do, you know, I mean.
Yeah. Yeah.
Sorry you didn't get the.
No, it's fine. It's been lovely listening to your experiences, in all honesty. Yeah.
Cool. But we're here if you have any questions.
Pretty much. Might be joining the
20 minutes passed too fast, honestly.
Yes, Alex, we can take this up now because we're here now for our last speaker session. And we're going to start with Pernilla Craig from Nathan Leporo's lab in the Bristol Robotics Laboratory, and she's going to talk about active interference for artificial touch. So Pernilla, take away.
Okay, I will share my screen. Hopefully this works. Just let me know if you can see everything.
Yeah, you can see it. Perfect.
Yeah, you can't see
Okay,
my. Okay, brilliant. So hi everyone. My name is Pernilla Craig. I'm a PhD researcher with the Farscope Centre for Doctoral Training, based at Bristol Robotics Lab. I'm sort of with the University of Bristol and the University of West of England, and funded by the EPSRC. And I'm supervised by Dr. Lawrence Aitchison and Dr. Nathan Lepora. I'm really happy to be here today to present my work entitled Active Inference for Artificial Touch, a biologically plausible tactile control method. So this work consists of implementing an active inference-based control system on a tactile sensing robot with the aim of completing an active localization task. So in this talk, I'm going to take you through the motivation behind my research, give you an understanding of active inference, as well as take you through the task I've implemented and present the main results and findings from it. So the main motivation behind all of this is basically to see whether we can create more human-like control in artificial touch sensing. So as humans, our sense of touch is sort of integral to the way we interact with the world. We sense pressure, texture, temperature, and using all of that information, we modulate grip strength, complete fine motor tasks, a whole host of different things. If you've ever tried to unbutton a jacket when you come in from the cold and your fingers are numb, you know how difficult it can be to complete even that simple task without the tactile feedback we're used to. And additionally, like a lack of tactile feedback is given as a significant reason for prosthetic limb rejection in upper limb amputees. And when our senses are dampened, our sense of touch allows us to continue navigating the world. If you think about trying to find a light switch in a dark room, even though you can't see, you're still able to complete this task. So then when we look at creating robots that are going to be interacting with the world, it's really important that we consider how tactile sensing could aid them in having successful interactions. To do this, we're looking at using active inference, which is a theory developed in neuroscience as a way to model the human brain and human decision making. And at its core, there's this idea that everything we see is seen through a lens of interaction and inference. So the brain is basically completely separated from the world, and it's only really able to interact with it through these different senses that we have, sight, smell, touch. And when we use these to interact with the world, we get sensory feedback. And this sensory feedback is sent to the brain, which can then use it to sort of infer and build an internal model of what it believes the outside world looks like. And then to test this model, it's believed we make predictions about how the world will change if we undertake different actions. And then after selecting the action we believe will result in the most favorable outcome, we see what effect this action has on the world and use that to update our internal models based on whether or not our predictions are correct. So if you again try and think about finding that light switch in a dark room, if you come in through the door frame, you might think, if I stick my hand out onto the wall next to me, there's going to be a light switch. If you were then to do that and not find a light switch, you would sort of reevaluate your hypotheses. Maybe you think it's higher or lower on the wall, further into the room. And you go through a sort of iterative process of actions and observations and updating hypotheses until you complete your goal. So basically, at its core, the control system we're developing here aims to use this inference process to inform decision-making and select actions that will lead to favorable outcomes. So specific goals of my study have been to implement this active inference-based control system on a tactile sensing robot and use it to complete sort of foundational localization task. So the task we've chosen is fairly simple. Chosen it to act as a sort of proof of concept to see whether this control method works and whether it has sort of merit for further study. And lastly, we're also looking to investigate different methods of driving behavior within this framework with a sort of secondary goal of removing human bias from the system. So lots of current and past methods for controlling robots use fixation points, so they use hard coding to influence behaviors. And this can all work really, really well. We've done lots of that sort of work in our lab. This sort of built off of or following on from some work that we've done with fixation points. But with this method, we're looking to see whether we can remove sort of all sources of human bias and see whether the robot is still able to exhibit what to an outside observer might look like an optimal control strategy. So the equipment we're using consists of a tactile sensor mounted on a robotic arm. So the robot arm we're using is a DoBot Magician 4 degree of freedom robot arm, sits on a desktop. And the tactile sensor we're using is a TacTip biomimetic optical tactile sensor. It has a soft deformable tip with a 3D printed skin. And on the inside of that skin, it has this sort of internal pin distribution. I don't know if you can see my pointer, but there we go. And no, I don't want that because I'm going to show you this video. So basically, when you poke the outside of this soft deformable skin, these pins on the internal surface move around, and there is a camera inside the sensor that is able to take videos or images in this case and track these pins and their deflections. And we can use that information to infer what sort of contact it's had with a stimulus. One of the things that's really great about all this equipment is that it's really accessible and sort of low cost technology, but still able to do some quite powerful stuff with it. So onto the task. As I said, the task we're looking at is a simple sort of where channel localization task in a known environment. So here we've got this robot or the sort of controlling agent looking to answer the question, where am I? So what we've got here is a hemicylinder. Again, I'll put this on. So we've got this sort of hemicylinder stimulus acting as the known environment, and we have our sensor starting at a fixed height above that stimulus, and it's able to move in this sort of lateral movement direction across the cross-section and vertically down a fixed distance to take tactile observations. So we define position states at equal sort of intervals across its surface. We have 61 of these states in total at 0.4 millimeter intervals. And doing this constrains our localization problem to one dimension. We build a model of what these states look like using a simple histogram classifier, taking a single tactile observation at each location and using the pin deflections that we see to build representations of each state, which we can then use to basically have a dissimilarity measure between incoming sensory observations and spit out sort of a best guess observation or observed state for which state the robot thinks it's in. Then to enable the robot to move around the system and gather data, we need to define a set of actions. And for simplicity, again, we define 3 different actions that correspond to moving left a single state, staying in the same location, and moving right a single state. So then when we run each test, we initialize the robot in a random state and allow it to take a series of actions and tactile observations to gather information and build up a belief about its likely location within that system. Then when its belief reaches a threshold level of certainty, say 80% certain that it's correct, it's going to stop gathering information and declare that it knows where it is and move on to the next trial. So I've said a lot about the robot knowing things, but how does it know them? That is the next sort of logical question that I'm going to briefly answer. Basically, all the information and understanding about the world this agent has, aside from this model, comes in the form of a set of matrices. So we have a set of transition matrices, one for each action, and these explain basically matrix transformations to explain how the robot's state will change if it undertakes each action. We have a prior belief matrix, and this sets the sort of initial understanding about the agent's state. So here, where our robot has no prior knowledge about where it starts, this is completely even across all states. And these last 2 matrices are what we can use to direct the behavior of the agent. So we have a confusion matrix, which encodes the confusion between the true actual state that the robot is in and the state that it's observed as classified by the model. And this utility matrix, which sets the desirability of achieving different final states. So if you have no bias in this matrix, it would be completely flat. All states would be equally as desirable as each other. But if you wanted to introduce some bias, you would give the values that were more desirable, sort of higher values within this matrix. And our agent, the way it's all set up, will tend towards sort of high information states, as in the confusion matrix, and highly desirable states, as in the utility matrix. So then, as I said earlier, one of our main goals is to investigate different methods of driving behavior, and we do that through testing our setup with different sets of confusion and utility matrices. So the model of the environment that we build at the start is not perfect. Some areas of the stimulus are more confusing than others, particularly in the center, where it more resembles a flat surface. And so when we build this model, we test it against sort of test data sets, and we find areas of confusion, which can be sort of passed along to our agents. So you can see in the middle here, we get some lighter values that sort of correspond to more confused states. However, these confusion matrices, whilst they demonstrate general trends, often have local minima, as you can sort of see, and these local minima can discourage the agent from exploring. So as a way to combat this, we test the control system using this model confusion matrix we have, and also a smoothed confusion matrix that we generated through averaging the confusion we see across several different models. And then for the utility matrix, we test again with two different setups, one that's completely unbiased and one where we scale the values in this utility matrix in line with the diagonal of this smooth confusion matrix to generate a sort of smoothed biased utility matrix. And doing it this way allows us to sort of highlight these higher information states, but without adding in a source of human bias, as everything comes from sort of gathered data, and there's no sort of human tinkering in there, which is what we want. So then when we test the control system, we tested with 5 different setups corresponding to combinations of each type of fusion and utility matrix, as well as a random choice baseline, and tested at 5 different decision thresholds, 75, 80, 85, 90, and 95%. And then, so this is basically what my robot looks like when it's doing its little pokey things. It starts in a random state, and then it sort of moves sideways or in whatever pattern it decides is best, and gathers more data until it decides that it's done. Just to give you an idea of what's actually going on. But then if we move on to the next slide, this sort of first graph I'm showing you presents the percentage of trials in each setup that reached a decision about their location within sort of designated trial length. So we just cap it at 20 trials because it's a lot of, or 20 observations, because it's a lot of observations. Both setups using the model confusion matrix that we have show sort of really consistently high percentages of localizations. This sort of random choice baseline we have here is significantly outperformed by everything we've done. But we also see a difference between the sort of smoothed utility matrix. So these are both for the smooth confusion matrix, and the smooth utility matrix results in much higher percentages of localizations than when we use the sort of flat utility matrix. You'll also notice that the model confusion matrix setups result in consistently shorter trial lengths, which is great, but it's only really great if we have accurate results. And if we take a look at the percentage of trials which localize incorrectly for the setups using model confusion, these sort of plateau at about 20%. Even for our really high decision thresholds where the agent needs to be 95% certain it's correct. And normally what we'd like to see is that being sort of on par. It needs to be 95% certain. You'd hope that 95% of the time it's getting it right. And when we look at these sort of smooth confusion matrices for both sets, this is kind of what we see. Like it comes down sort of in line with our decision thresholds. And then if I move on here, we can also see the average error for each setup. So just going to point out that even with our sort of most inaccurate set of conditions, we still only see an average error of about 1.144 millimeters, which is on par with results we've gained in the past using other methods. And it's pretty good, all things considered. And overall, basically, we have a trade-off here between our two different types of confusion matrices. So with the model confusion, we're able to localize really quickly, but at the expense of accuracy. And with the smooth confusion matrix, we're able to gain a high degree of accuracy, but it takes longer to localize. So there is a little bit of similarity at lower decision thresholds here. Quickly moving on. So now one of the really cool things that sort of comes out of this, and we can see from our results, is our setup is able to overcome incorrectly classified data. So throughout these tests, we use finite horizon iterations to consolidate information after every 3 actions. So we've done a lot of simulations. This is basically just what shows best. So as a result of this consolidation process, our agent's able to ignore or move on from confusion it's had in the past. So in this graph, we can see the agent's belief it has about its location at each time step corresponding to each action and observation it's taken. And in this first set of actions that we have here, it receives directly contradictory information. So after action 1, it believes that it's in this state here. And then after not moving, so it should still be in the same state, it receives an incorrect reclassified state. And you can see that sort of represented by this confusion between this sort of color discrepancy here. And then again, after not moving, it received another incorrectly classified observation, which is not fantastic. So this has been sort of reaffirmed by its previous observation, which is still really confused because it's in direct contradiction from what it's seen in its observation before that. But then when we consolidate all of this information, we pass it on and do a second iteration, we're able to, through a series of correctly classified observations, actually come and not only sort of localize correctly, but we're also able to increase this understanding a little bit that it might have been incorrect in this first position, which is good. And then finally, we've got the trajectories that are chosen by the agent. So here we see the results for the sort of smooth confusion matrix and unbiased utility matrix. And we're able to see this emergent trend where they're moving selectively towards the edges, where they have these higher information states and away from the center of the stimulus, sort of in the middle of this graph, where it more resembles a flat surface. But what is quite cool is that if we look at the comparison when we use these sort of biased, smoothed utility matrices, is that we're able to amplify this behavior, the agent moving much faster towards the edges, which is likely why these trials had a much higher percentage of localizations and generally shorter trials. So to conclude, we sort of successfully implemented this control system on our tactile sensing robot, and we've achieved a localization task to a sort of high degree of accuracy. We've also demonstrated the system's able to overcome misclassified data, successfully localize, and we've shown that it's able to exhibit sort of emergent behaviors that might be akin to optimal control strategies, which is great as we look towards more complicated tasks and moving into unstructured environments. So hopefully I haven't gone too far over. Thank you so much for coming and listening to me, and I'm happy to answer any questions you have now or by email later.
Thank you, Pernilla. We would have time for one question. I don't see any right now in the from the audience, but I have one question here. How does it actually really indicate that it found its own right spot? So is it in the data, or is it really an audible sound that it makes? Okay, hey, I found my spot.
It doesn't make an audible sound. It is just within the data, although I do quite like that. And I might, although I don't know if the people I sit next to in my lab would be particularly happy if I introduce beeping.
Or a flashing of the light or
flashing
something. That
lights.
would be also
Yeah.
good. No. Yeah,
Yeah,
cool.
no.
It was really cool. I like that talk. It's really nice. Thank you very much. And if I would, due to time, I would say we now go on. And if you have any questions, please remember we have our Discord channel where you can always ask any questions left here. And now we go on to Ahmed Mohamed from the Ames lab of Joseph Najam from Penn State. And he's going to talk about the design of biomolecular neurostore circuits for bio-inspired control. So Ahmed, it's your turn here.
Yes.
The floor is yours.
Let me share my screen. This one. And I hope you all see my screen.
Yes.
Okay.
Perfect. Now we still see all of your slides.
Give me a second. I'll fix that.
No problem.
Oh, does that?
I think that should fix it.
Do you guys see
my
my presenter view, or is it? Oh.
No, we see still your presentation with your PowerPoint, not the presenter or anything. We just see your PowerPoint with all slides and currently the main slide.
Oh.
Can you extend display or like move the slides there?
Let me do that.
Yeah. Yeah. Into the presentation.
That didn't work.
And then go back to Zoom too and stop screen.
Sorry about that.
Oh, now we see it.
Oh, they see it?
Now you see it?
Oh, they do.
Now we see it.
Somehow.
Somehow. I don't know how, but we see it now.
Cool. All right, everybody. Sorry for the technical difficulties. I am Ahmed, and I'm here today with my collaborator, Ashley Liao, and we're here to present to you our shared work, design of a biomolecular neuristor circuit for bio-inspired control. To start off, Ashley's going to motivate the talk. Ashley, floor is yours.
Thank you, Ahmed. So I'm Ashley, and I'm from the Biohybrid and Organic Robotics group. And our group is really interested in soft bio-inspired robots, but also biohybrid robots. And both of these systems really need a robust, adaptable control system, in which there are also like two avenues that we can try to pursue this. One would be developing a bio-inspired controller, and the other would be developing a biohybrid controller. However, implementing either type of controllers can be pretty challenging for both soft and biohybrid robots. For bio-inspired control systems, they're often composed of rigid synthetic electronic materials that may not be biocompatible. In addition, they usually have high energy needs and complex computational implementations. On the other hand, biohybrid controllers tend to have a complex fabrication process using specialized equipment and clean rooms, as well as the handling of fragile cells, which can be really difficult to maintain. And this led us to our collaboration with Penn State, because we really had this major question that was motivating all of this work, which is how can we engineer a biocompatible soft controller that produces bio-inspired dynamics? So for these types of controllers, we really want to be able to capture neuron dynamics. And by neuron dynamics, we want to be able to produce an action potential. So for neurons, their action potentials are generated by the ion channels within their membranes. And then within these classes of ion channels, there are two major players: the depolarizing sodium channels and the hyperpolarizing potassium channels.
So our lab proposed the use of a neuristor, which is by definition a synthetic device that exhibits the firing generation capabilities of biological neurons. And neuristors have been built in the past, but what's different about this neuristor compared to the silicon-based and CMOS-based neuristors that have been built in the past is that it's soft, biocompatible, scalable, operates with ionic mechanisms, and operates in biologically relevant timescales. So we've drawn inspiration from biological neurons, and we've conceptualized this neuristor to have a depolarizing memristor that emulates the depolarizing behavior of sodium ion channels, and a hyperpolarizing slow memristor that emulates the hyperpolarizing currents of potassium ion channels. So we've constructed individual memristors in lab. We haven't constructed the full device, and to construct a memristor in the lab, what we do is we deposit a droplet of aqueous solution in oil. The aqueous solution has salt, lipids, and alomethicin peptides dissolved in it. Because of the lipids, hydrophilic heads and hydrophobic tails, the lipid liposomes diffuse at the interface between water and oil, and they form what we call a lipid monolayer. And once we get those lipid monolayers closer and in contact, they form at their interface what we call a droplet interface bilayer, similar to a biological membrane. So due to alomethicins voltage sensitivity, once we apply a transmembrane potential between the two droplets, what happens is alomethicin inserts into the bilayer, creating an ion channel, which allows ions to move from one droplet to the other, depending on the voltage sign applied. So this is just how we construct a single memristor. But when we conceptualized the neuristor, we came up with a model, computational model that captured the neuristor dynamics. And I'll show later that we've tuned the model parameters to get various firing patterns and frequencies. And we envisioned that those parameters in the model would correspond to lab-related parameters that would allow us to get those different firing pattern and frequencies in lab. And finally, we really want to show that this neuristor can be embedded in actual bio-inspired control circuits. So what we did is that we did in simulation put the neuristor circuit in a larger mutually inhibitory central pattern generator circuit, and we compared that to one that's constructed using neurons in simulation. To start off with the model, we constructed a neuristor dynamics, a neuristor model that is inspired by the Hodgkin-Huxley model, where the membrane potential is described by a current balance between the stimulus current, the current generated by the depolarizing memristor, current generated by the hyperpolarizing memristor, and the currents stored at the bilayer, the membrane. And for the for the currents generated by the ion channels, it's a simple Ohm's law G times V, where the conductance term G is comprised of the conductance of a single channel GU multiplied by the number of open channels per unit area N, and multiplied by the area A0. And to capture the number of open pores, we've used a first order state equation that's nonlinear, where the time constant term is exponentially dependent on voltage, and the number of steady state open pores is also exponentially dependent on voltage. If we simulate this system of state equations, what we get is an action potential like spike that is an all or nothing response similar to biological neuron, and it has a refractory like period at the end of it. So what's really interesting about this is that we can tune the parameters models to get different firing patterns and frequency, but what's even more interesting is that we can correlate some of the parameters of the model to actual fabrication parameters in the lab. For instance, the membrane capacitance Cm can be related to the interfacial area highlighted in red, which is controlled in the lab by the droplet size. Similarly, we can control the speed of the system by changing the peptide concentration, lipid composition. And we can also control the currents generated by each of the membristors by controlling the steady state number of open pores, which is in the lab controlled by the salt type, salt concentration, and peptide concentration. For the coming slide, I'll be showing that we can tune some of the parameters to change the constituent components of this action potential. So let me go into the constituent components of this action potential like spike. So if we have two membristors connected in parallel and we turn on an externally applied current source, what's going to happen is, because there isn't any channels inserted and it's just a bilayer being charged, the membrane potential is expected to rise linearly until at some point it's going to reach the insertion threshold of the depolarizing memristor. And due to the threshold, the membrane potential reaching the threshold, alomethicin is going to insert into the bilayer and form pores. And the number of open pores is shown in the dashed line, and it's rising. And due to the ion gradient imposed, ions are expected to move from the negative terminal to the positive terminal, further increasing the membrane potential upward until it reaches the insertion threshold of the hyperpolarizing memristor. We really designed this hyperpolarizing memristor to be very slow so that its impact isn't very is not dominating at this stage of the action potential. So the membrane potential further rises until it reaches, becomes very close to the reversal potential of the depolarizing memristor. At this stage, the number of alomethicin channels start to withdraw from the depolarizing memristor, which is shown in the left by the different blue and red lines. There's just a slight difference in slope. It's not very visible. But from there, due to the withdrawal of alomethicin channels from the membrane, the depolarizing membrane, the hyperpolarizing membrane starts to dominate the effect and pull the membrane potential downward due to its opposing ion gradient. And this is what happens with stage 5. And because the hyperpolarizing memristor is designed to be very slow, it's even after passing the thresholds of both membristors, it withdraws very slowly from the membrane, but allows the membrane potential to be pulled further downward to go even below the starting point, which sort of emulates the refractory period-like effect that we see in biological neurons. And at this point, for both membristors, channels would have fully exited. And from here, we charge the membrane again, and the cycle continues as long as we have a stimulating current source. So this describes the 6 constituent components of our action potential like spike. And in the coming slide, I'll be showing how we can control some of those constituent components by changing some of the model parameters and the corresponding lab parameters. So let's say our target is to get a fast firing spike. So for that initially rising part, when we connect the stimulus current, what in the model, what controls that slope of that initial part is this parameter A0, which corresponds to the interfacial area. So the smaller the interfacial area, the smaller the capacitance, the faster the system is going to charge. So if you want to pick something from the right choices to get something that charges fast, we pick the smallest set of droplets. And to get even faster firing, we would love to have something with a depolarizing memristor that inserts at a low threshold. So to get a lower insertion threshold, we changed the parameter N0D in the model, which in the lab would correspond to a memristor with high L-amethicin concentration. So we select the highest of all the choices. And not only we can control the threshold, we can also, for memristors with similar thresholds, we can control the speed at which the membrane potential increases. And to do that, we can control the membrane composition by changing the membrane, by choosing membranes with different viscosity. The lower the membrane viscosity, the easier it is for channels to form in it. And so we pick the membrane with the largest fluidity or the least viscosity to get the fastest response. We can do the exact same analysis or selection process for the hyperpolarizing memristor, and we can choose a hyperpolarizing memristor with the lowest insertion threshold, which corresponds to the highest L-amethicin concentration. And we can also control its the speed at which it pulls the membrane potential downward by changing its membrane composition, similar to the way we did with the depolarizing memristor. So this is one way we can generate a relatively faster spike in concept, but the question still remains, even if we can produce different spiking patterns and frequency, can we really use this neuristor circuit to embed it in an actual controls, bio-inspired control circuit for bio-inspired applications? Ashley?
Yep. And so because we were able to model this action potential like spike and tune its properties, this got us really excited about the potential to develop a bio-inspired soft controller. But we still needed that proof of concept to really see if this is a viable solution. And so what our next steps were to compare the neuristor model with Izaykovich type neuron models as well in simulation. And so we set up the neuron model in a mutually inhibitory circuit configuration that was composed of 2 excitatory neurons and 2 inhibitory neurons. Similarly, we set up the neuristor model that Ahmed just presented on in a similar fashion with 2 excitatory neuristors and 2 inhibitory neuristors. Unsurprisingly, the neuron models produced a alternating firing behavior, which is what we would expect from a mutually inhibitory circuit. But what really got us excited was that the neuristor model also produced this alternating firing behavior. Now, while we do see that the spiking dynamics are not exactly the same as the neuron model, the fact that we were able to tune our model and still produce this alternating firing behavior shows a lot of potential that we may be able to further modify this neuristor even in the lab to more closely model even the individual spiking that we see from neurons. And so to conclude our presentation, we have presented on a synthetic biomolecular device called a neuristor that can produce bio-inspired neural activity patterns. We also showed a computational model of the neuristor that can be used to identify relevant experimental parameters that can alter our fabrication process to tune these neuristors with more neuron-like electrical dynamics. Furthermore, we also used this computational model in a mutually inhibitory circuit and found that it was able to demonstrate the alternating firing behavior that we would like to see. But because the spikes weren't exactly the same as the neuron on the timescale, we do need to further optimize our parameter tuning process so that we can also more efficiently match that spike and potentially additional neural profiles. So I would like to end by thanking all of our co-authors and collaborators, as well as acknowledge all of our funding sources. And we would be happy to take any questions at this time.
Thank you very much. Perfectly on time. So I will just check if we have any questions. I have a question directly because I find it's a really interesting setup you got there. And one thing that I wanted to ask is these ion channels, as you said in the starting foil slides you presented, you said that they self-assemble out of the alamethicine, right?
Yes.
And you'd say that they actually can be in slide 11. It was pulled out from the out of this membrane, this artificial membrane, and do they actually deassemble again into its components, so into this alamethicine? So would they change actually the alamethicine concentration there, or are they staying as an ion channel then and can be reintegrated into the into this?
So if I'm understanding your question correctly, you're asking if once we remove the applied voltage, does alamethicine fully withdraw from the membrane, or does it just like does it withdraw as full ion channels, or does it fully separate as single? That's a good question. According to my knowledge, because the literature on alamethicine is usually very old, and there hasn't been much done in imaging, at least that I'm aware of. But what I know is that alamethicine exits as a full ion channel out of the membrane. But there has been papers that show that they just de-oligomerize or just separate the ion channels after you remove the voltage. It might be a different setup, but there might be more than one way they do that. But my own, just to conclude it, my own intuition is that they come out as channels out of the membrane.
Okay, thank you. We have one question from Hillel.
Yeah, hi. Lovely talk. Thank you for the beautiful work. You focused on temporal matching, which I think is very important. What about spatial issues? How small or how large are these? I'm assuming that the maintenance of the gradient depends on the changes of concentration. And if you make the droplets too really small, then it's going to be hard to maintain the concentration gradient. In neurons, you would have pumps of some sort that would revitalize that. Here, if you have a small enough droplet, then each of the action potentials could significantly change the concentration, and then your concentration gradient begins to break down. So I was just wondering what you thought about in terms of spatial miniaturization and scaling up.
That's a great point. Yes, so that is true. Once you have, because of the changes in the ion gradients accompanied with each action potential, if it keeps firing for a long time, you start losing this ion gradient. For the current work, we haven't really, we're not really tackling this issue, but we might be considering adding pumps maybe, but also increasing the droplet size would help. So we might be adding constraints on the droplet sizes for applications that last for a specific time. So that's one way we were thinking about that for the current setup. But we do have a fire emergency.
Have your attention, please. Fire emergency has been reported in the building. Please leave the building by the nearest exit.
Well, that's a great exit. You did it. You engineered it, right? You were worried about more questions.
We can answer any more questions on the Discord. Unfortunately,
yeah, that I wanted to suggest this. So please leave the building and stay safe.
But thank you for your questions.
Thank you so much for the question.
Yeah.
Thanks for your talk.
Nice. Great talk. Then I would say, luckily, Fabian is not in this building. We switch to the next talk, who will be presented by Fabian Mida from Barbara Mazzolei's group in Istituto Italiano di Tecnologica. But I think Fabian is not presenting.
I will present.
Okay.
I'm Goffredo Giordano. Nice to
Hello,
meet you all.
Goffredo Giordano.
Okay, so can you see my screen?
Yes, perfect.
Okay, nice to meet you, everybody. Good evening, I'm Goffredo Giordano. I'm co-author of this work named Underwater Light Modulators, a study on iridescent structure of the seagrass Posidonia oceanica. I'm glad to be here at the Living Machine conference. I'm from the Bio-Inspired Soft Robotics group led by Barbara Mazzolai at the Italian Institute of Technology. And first of all, you will ask yourself why this more biological study is on living machine. Our idea is, first of all, to try to learn from nature, and in this case from the light modulators like plants. Okay, and to take inspiration in order to merge with the technical field like material science or soft matter physics and chemistry, and to merge them into a smart machine, a soft robotic, for example, manipulator that can be endowed with material intelligence, mechanical intelligence, or learning intelligence. In this talk, I will speak mostly about material intelligence as regard properties that we can integrate into the soft host material of a soft robotic manipulator in order to obtain, for example, chromogenic soft sensor without electronics. So our main loop, our framework is to obtain in the future some autonomous soft robot that can be proprioceptive. So they can interact with the environment, acquire information, elaborate their data, and define control in order to come back to the environment and explore this. To do so, in particular, we need mechanical intelligence, and we have some example of reconfigurable shape system like an octopus tentacle or a tendril-like system. But in this talk, I will speak more about material intelligence as regard optical properties driven and leaded from natural system in order to obtain iridescent sensor. In particular, why iridescent sensor? Because we envision that avoiding electronics and flexible electronics that still now is not that common and reliable, we can instead change the paradigm and integrate into a grasper some smart solution in order to visibly interact with the human or with the environment itself in order, for example, to communicate whether it is achieved or not a grasping. And to do so, let's start and go deeply on the work that we presented here at the Living Machine conference. There are several examples from the from plants and animals on organs and structure that arranged in the evolution in order to modulate light, just modifying their refractive index or the thickness of this arrangement, multi-layered arrangement. And the optical counterpart and technological counterpart are very well known, are photonic crystal. In our study, we will analyze in particular a one-dimensional photonic crystal, so a distributed Bragg reflector that diffracts light because of its periodicity in the refractive index and into the dimension of the multi-layered stack in order to interfere and modulate light accordingly. Actually, our biological model is this leaf blade of the seagrass Posidonia oceanica. We don't have answer to the main biological question why this system arranged in this way. But we started analyzing from the scratch the color changing along the leaf blade. And you can see how from the base to the upper apex part of the leaf blade, the color change suddenly from the white part of the basement up to the greenish part in the upper part, in the upper in the tip, let's say. And then we thought to analyze spectroscopically whether there is something that is behind the cuticle, the skin, let's say, of the blade, of the leaf blade. Analyzing with a very simple spectroscopic experimental setup, let's say, and integrating an angular dependency in order to understand whether there is iridescent properties or not, we acquired some data, and you can see how in the bottom right, the reflection spectra of the base show a large photonic stop band, so a wide reflection along all the visible spectrum, appearing then white, while the lower that appear yellowish show two photonic stop band, one in the near infrared region and one in the greenish yellowish part. Whereas the medium and upper apex show just a photonic stop band in the near. Then we started analyzing optically and more in depth with the scanning electron microscopy the arrangement of the structure. And whether with the optical microscope we didn't get some crucial result, you can see in the bottom right in the comparison between the upper picture at the SAM and in the lower, that and in particular in this picture, how the arrangement of the organic part behind the cuticle is mostly organized as a multi-layered structure. From the dry leaf, we can see that there are some empty spaces. Then we hypothesizes that whereas because the seagrass live in the seawater, so in a wet environment, maybe perhaps this invagination can be filled by water. Then we developed a model based on the transfer matrix method on a computational model, analyzing a one-dimensional planar distributed Bragg reflector with the dimension and the refractive index parameter determined by the acquired picture and the refractive index from the literature, in order to reconstruct the optical spectra. The computing model didn't get the second order photonic stop band that I showed you before into, I can come back here, into the black line. While this, and that is the structural color that we wanted to look at. But we can, with this simple model, we can analyze that with an optimal configuration, the iridescent behavior can happen. So it is angularly dependent. So the reflection spectrum changes along a wide range of angle. But there was still this open question about the second order photonic stop band. And I will show you that we analyzed, first of all, experimentally, the spectroscopic analysis as regard several regions from the basement to the lower, to the medium and the upper, looking at these spectra are angularly dependent. These spectra are angularly dependent, especially in the lower part, while in the base, medium and upper not. And this angular dependency remind us that maybe there is an iridescent behavior, and then there is a structural coloration. Then we computed again in another model, the model, modifying it a bit. In fact, if you look at if you remind the top right picture at the SAM, you can see that the geometrical arrangement of the photonic DBR is not planar, but is mostly we modeled as a trapezoidal system. And modifying the angle at the between the two planar DBR, you can see that the second order photonic stop band appear, and there is a blue shift of the first order as in the one-dimensional planar DBR, while in the trapezoidal DBR appears also a red shift of the second order photonic stop band, resembling the result that we obtained from the experiment. Then I can show you some of the preliminary results that we achieved, and we go toward, let's say, the technological point. In fact, that is not based on the same behavior of the plant-based system that I analyzed before, but it is mechanically driven. So we have two dielectrics that can be two polymers like PDMS and polystyrene, and a copolymer polystyrene block polyisoprene that can be wrapped around another support, and you can see very let's say a bit of evidence, this structural iridescence that is more clear at the optical microscope, whether when there is a mechanical stretching, the color change. So you can engineer your, let's say, artificial skin in order to change the color, whether, for example, there is a damage threshold on the strain applied. To conclude, my group and my collaborators, also Fabian and Serena, take inspiration from this work started from Fabian, that study in depth the behavior of the leaf of the plants. They work on a biohybrid system for energy harvesting, and maybe you can have a look, obviously, or you had a look before about their work to harvest energy. And that's all. Thank you, everybody, and I'm open to all your questions.
Thank you very much for a nice talk. Are there already any questions? If not, I would like to ask a few, because so you showed us in the start a picture of these leaf blades, right? Where you see that it is more brown, and here, for example, there also you see that is near to the stem is more brownish, and then gets more greener, light green, and then goes into a dark green. So is that only structural, or is it correspondence with a chlorophyllc concentration?
Yeah, that is a very good question. And indeed, if you look at its structure, I mean, behind this, let's say, invagination, there are chloroplasts. So there is chlorophyll. Most of the let's say, acquisition also that we performed show that there is obviously a contribution of the absorption due to the chlorophyll that is into the leaf. We concentrated, we focused mostly on this lower part of the leaf blade because we evidenced this, highlighted this difference of coloration. And we then asked ourselves, is there a changing of color because there is another arrangement or distribution of pigments inside, or it's a structural color due to some, let's say, nanostructure that can evolve in this way. And, as far as we know, biologically, there are no answers to this question, and we just reported let's say a fact.
Yeah. Okay. Cool. Thanks. Just one follow-up question, if there isn't any. So, is this iridescence? Is this actually a evolutionary advantage? So, would it be like because if these high lighter in colored structures are near the base, right? So the big leaf and the leaves are like what 2-3 meters long, as I gather. And so would it be that they distribute the light a bit better to these points where they get not so much light because they're near to the ground? Is that
yeah, you're right. Obviously, all of us maybe encounter once in their life this seagrass because it's on the basin of most of the sea. First of all, in the Mediterranean Sea. Evolutionarily speaking, we don't know, and maybe we have also to not look with the, let's say, human eye, because we need to understand what is the interaction between the ecology of the system at the basin of the sea and why they changed this color. Obviously, maybe on the top part there is more chlorophyll because maybe is more close to the surface of the water that can harvest light from the sun. On the bottom, maybe we don't know, maybe there is some communication skill, but it's really something that is a speculation, and we don't have an answer at this question. But it's a good question that many biologists maybe ask themselves.
Thank you. So if there aren't any questions anymore, this then concludes our last presentation section and session. And I will give over to Alex to talk us through the next parts.
Excellent. Great work, everyone. I am dropping the polls in the chat again. So vote for best talks, best short talk, best long talk, and polls will close in 20 minutes. And we'll tally that up for our award ceremony in 40 minutes. So the next few steps of the program is just, we got a short break, and then we'll put everybody in breakout rooms again for another discussion. The focus that we picked for this last discussion is on diversity, equity, and inclusion. And I invite people to think about this in the context of our own conference, the field in general, their own individual disciplines, and kind of, I mean, we're not gonna solve things here, but kind of starting discussions and thinking about ways to improve things. So with that said, we'll go into a short break and I'll form some breakout rooms. And whenever you get back, then you can start those conversations.
And hopefully Vicky and her group can come back, because I think the alarm stopped in the picture that we see. No more flashing lights. Hopefully it will come back. See you soon.
Oh, one other comment for the kind of leadership committee. We're going to get into our own breakout room to kind of discuss next year's living machines and the poll results.
Alex, when are we going to do that? Alex,
yeah, we're gonna we'll take our own quick breaks, bio breaks, whatever you need, and we'll get into that breakout room very shortly.
Real soon. Okay, got it. Thanks.
Hey, Alex, we're back. We're live. What part of the program are we in?
We got a short break, and then we're going to do the diversity, equity, and inclusion. Vicky, I was wondering if you could hop on a separate account for a leadership meeting.
Yes, yeah, let me go find a place in the hall and I'll be on in just a second. Also, FYI, the chat is currently disabled on Zoom. I don't know if that was intentional.
Yeah, because I was also gonna send you if I need to go to that specific channel and where is it?
I am making it.
Okay, send me a link. I'll be out in a bit.
Log into this main thing. It's just going to be a breakout room.
Gotcha. Okay, see you in a jiffy.
Hey, Alex, quick question. Sorry, do you want me to pause or stop recording for a little while? I don't know how that works.
Yeah, go ahead and stop it.
Okay.